directly from the people who is promoting it. I think that's the best way to learn things. You listen from the horse mouth. That's what it's like. So, Muruganathan sir, thank you so much. Right here at 8:30. I was late. So, but I made it a point to start by 9 o'clock sharp. Thanks for all you are here. So, floor is yours, sir. And you can go to 9:45 because we have other programs lined up. And they were exposed to digital droplet PCR yesterday by other speakers. And they know a bit. Probably they would not have experienced in the lab. But theoretically, perhaps most of them are quite aware of the technology. And I'm sure you will be in the comfort zone and also maybe in the comfort zone of exchanging knowledge.
This is online. So this goes to online. We have online views and we have policies. Good morning. Good morning and
Speak in the mic.
yeah.
What do you call that? Condition.
Okay.
This is for the audience. This is for the online.
Okay. In molecular immunology, so I have a PhD from Christian Medical College, Vellore. So more into diagnostics, but or human diseases and infection. But then my interactions with people like Dr. Kumar also made me learn a lot from various other aspects, plants, animals, microbes, everything. So over a period, we have also accumulated a lot of scientific understanding and also as Eppendorf, we are more into the sample preparation devices. We also complement products from other major vendors like Stilla Technologies. It's actually a French company having a base in US who have come out with the digital PCR technology almost a decade ago, and they are very strong in their own technology, which I will present you today. The strength of the, I'm sure you would have already gone through the basic principles of digital PCR, but then we present ourselves as a premium supplier for users who are interested in the multiplex part of digital PCR. So that's where I'm presenting you today. Of course, it will start with some basics and I will proceed to the details. So Eppendorf India partners with Stilla Technologies in India to support our customers, of course, to expand our offering to customers who are into molecular biology. So, Eppendorf offers a range of products for PCR amplifications. We had real-time PCR earlier, but then discontinued, and now we support the digital PCR. Very basics of PCR. So we start with the target. We have our specific primers that are used for specific amplification of target sequences, and it goes through the 3-step cycles or a 2-step cycle of annealing, denaturation, annealing, and extension. The components are known. It's a polymerase-driven process which accumulates or amplifies the target sequence and helps in understanding further downstream about the target sequence, about the presence, a lot of applications. But the conventional PCR has its own challenges, and that's also the reason why we have evolved to the next levels of the real-time PCR or the qPCR, and now the digital PCR. not going into the details, but to understand the chemistries, the fluorescent chemistries, it will be interesting because when we amplify the target, the tagged probes releases the fluorescent dye, and the quenching effect is removed, which allows the system to detect the fluorescence. So it's a way of indirectly measuring the amount of amplicons which are being produced by the enzyme. And this graph has become very familiar during the COVID times because even the layman understood what RT-PCR is, and the CT values are like very common terminologies. So to construct, basically for quantification, this was the design of the qPCR was mainly for quantification, but however, this also was had become a tool to identify basic screening of target sequences. And over a period, we also, if you have already used the qPCR, you also would have understood the challenges in qPCR. And of course, it has its own applications. We have applied it in several areas, such as quantitative analysis of the targets, the gene expression, especially the relative quantification, where we compare between a normal and infected or a normal and experimental subjects. We also have used them for absolute quantifications. However, absolute quantifications were dependent on a standard curve or a calibration curve that we use with known standards. So the known is a big question when it comes to the exact quantities that we estimate or quantify. And for pathogen detection, it's a very common, very interesting, easy to use technology. And it has also been consistently used for mutation screening, not the detection part, because the detection has to happen in the sequencing. So it has used, it's been used in the screening part. And of course, it has been used in multiplex, but with its own challenges. Advantages, of course, very short runtime. There is no post-PCR analysis. You just run, complete, you have the graphs, you have the CT values, and go ahead. And the limit of detection is more sensitive than the conventional PCR, with a dynamic range of 10 log, 9 logs, or 5 to 9 logs, depending on the system that you use. But the challenges remained widely because we were dependent on standards and references. So unless otherwise we have a standard to make a standard curve, we don't have the right quantities. And the standard curve is a question of who decides the standard, how validated is the standard, whether the standard had any inhibitors in it. So a lot of questions around the standards. And if you look at the MIQE guidelines, there's a big list of requirement for the standard evaluation. And the PCR efficiency by itself, because it depends on various factors. The primers that we design, the inhibitors that we have in the source, which cannot be removed, and the quality of RNA. Absolutely, sorry for that. So a lot of PCR efficiency questions in this. And of course, when we looked into the quantification, the requirement is also a single copy. And this is where the qPCR had its own challenges, because the PCR efficiency or inhibitor can provide you a false result. And of course, when it comes to multiplex, then the competitive inhibitions, because we have the reaction happening in the same tube. If you have multiple targets, the targets are competing with the enzyme, and the targets are competing with the available nucleotides. So if one copy, one of the targets is at a very high concentration, very high copies initial, then it consumes more because it utilizes maximum of the nucleotides as well as the enzyme activity. Wherein if in the mix you have another target which is at a very low copy number, so it will take an inhibiting factor during the multiplex. So this is how we do the multiplex. So you have the same PCR mix with 4 different targets, and assume that the variant 1 here is in higher copy number and the variant 4 in a very low copy number. So what would happen is, in most cases, depending on the amount of targets that you use, the variant 4 will have its own influence. It may not be detected as efficiently as your variant 1. Yeah. And of course, here the low copy challenges are many, and the efficiency is a big question, whether, and this is also the reason it has not been very successful when it comes to the qPCR. And as you already are aware, the digital PCR technology, so here the master mix is dispersed or partitioned into multiple droplets, and the amplification is done in these droplets. So the idea is divide and amplify. So you just divide all the 25 microliters of your reaction mix into several thousand droplets, and now each droplet is capable of amplifying on its own. And ultimately, when you just read the final amplified product, and you know how many of these partitions are having the fluorescence. So it's also based on fluorescence, similar to the qPCR. And if you ask me, if you have already a kit which is optimized for your qPCR, you can still use, and you can optimize it for the digital PCR. So this allows the user to read exactly the number of copies based on the number of partitions that are lighting up during the screening process. And here you have the possibility also to read signals using different wavelengths. So if you are using a multiplex with multiple dyes, then you can also read those multiple dyes differently, but also you can merge, overlap these results and see which are the drops, which are the partitions where you will have multiple targets also. So this allows users to really get the exact quantities in their amplified products. So the advantage here in the system, the Stilla Technologies offers a 6 multiplex system. So wherein you have 6 different channels can be used. And say a typical would be FAM, VIC, Joe, or so all those dyes, with additional infrared dyes like Psi3, Psi5, and other dyes. So now if you can actually do a multiplex irrespective of because here you don't need to really worry about the inhibition, because the inhibitors are also partitioned somewhere. So they don't inhibit all the enzymes or all the reactions. So you are trying to minimize the effect of inhibitors by partitioning them or trapping them into certain partitions. And this also is increasing the efficiency. A typical comparison of qPCR and the digital PCR, what are the advantages? So if you have to do a gene expression in your samples using a qPCR, then you need to go for a relative expression where you need to have a housekeeping gene, you need to have a target gene, housekeeping or a reference gene. But here you don't need, with the digital PCR, you don't require a reference. You can straight away use your target to quantify. But if required, you can also use the relative methodology adapted here to compare between samples. So that's the additional advantage. And the precision with which it detects mutation is very, very high, because the detection rates theoretically is above 1%, and here detection rates are as low as 0.001%, which provides a 100-fold increase in the precision. As I already touched upon the amplification efficiency and also the inhibitor tolerance and the reliability, because you can do multiple replicates to see how reliable your experiments are. And in qPCR also you can do, but then there, if you look at the requirement, real requirement of the number of replicates according to the MIQ guidelines, you will really be surprised that most of the requirements for a validated assay would require 6 replicates, not 3 or not 2, because that's the general tendency. There are multiple products available in the market. There are chemical partitioning, which uses chemicals, and emulsions, which can create droplets. There are also physical partitioning, where the samples are dispersed in a plate format for amplification. Each has its own pros and cons. Getting into this at the moment. But then we need to also look at what are the differentiators, how when you want to choose between different technologies. Of course, all of them are offering a digital PCR, but what would be interesting as a user, as a buyer, is the cost, of course. That's the first primary point. But the ease of use, how easy it is to use, how complex. So reduce complexity in the sample preparation, that's very important. And the throughput, because it depends again. There are labs which are using several hundred samples per day, but there are also labs which are using a few samples, maybe 3 or 4 samples in a day. So we need to also see how this throughput is handled, and hands-on time, how much time it requires to complete the experiment to have the results. Easy to maintain, and of course the software and analysis part, multiplexing capabilities, because you are not going to spend the in a multiplex assay will be very less or efficient, cost efficient than doing a single plex assay. So that's something which we can also look at. And the partitioning dynamic range. So in case if it's an unknown sample and you don't know what concentration it is, it could be 100,000 copies or it could be 100 copies. So whether this range, if you have this range of unknowns, will it pick up? Will it show that this is the range within which the targets are available? So these are things that we need to differentiate. And also the partitioning power of the digital PCR itself. So how efficient the partition happens? Is it, most of them are automated. Some of them are instrument dependent, some of them are reagent dependent, and some of them are physical property dependent. But it depends, it also needs to ensure the partition is of the same volume, same shape, same size every time. So every time you create a partition, it has to be the same, so that the reliability becomes more in terms of repeatability as well as reproducibility. About Stilla, as I said, it's almost a decade-old technology, came out of the university in France and then adapted by companies, and the company is almost 10, 12 years now. Very simple steps with respect to the instrument. The instrument is a chip-based instrument. So you can see there are several variants of chips. So you can have a sapphire or a opal chip. The sapphire has 4 samples per chip, and the Opal will have 16 samples per chip or 8 samples per chip. We also have a Ruby which is 16 samples. So this is the master mix for the chip. It comes filled with the oil, and you just need to add your sample and then it's ready to go. The amplification, so this is the geode is actually the system which allows partitioning as well as amplification. So partition followed by amplification as a single step. So there's no need for a manual interference during this step. So this also allows a very high level of confidence in the system, supported by a pump, a pressure pump, which allows the partitions to fill. I will show a picture in the following slides. And then the reader, which is basically a scanner. So the scanner is more digital pixel-based scanner. And the advantage of the pixel-based scanner is that you can also now have a lot of digital compensation. So the digital compensation is if you have one wavelength and if you have two dyes in the same wavelength, differed by a few nanometers, that can also be read or differentiated very easily and by the digital means. And of course, the software which can provide you all the analytical informations. Mixes, of course, the NICA system offers two types of mix. One is a 5x and one is a 10x. One for the cyber green or the EFA green dice, and the other one for the TACMIN dice. But also the dice, the master mix comes with a fluorescein to provide information on the visible quality control. I will come to that. Just remember fluorescein and visible quality control. I will come to the next steps there. And you will also see the 5x and a 10x mix, because the 10x mix allows you to increase your template or increase your template DNA. So you can increase the volume more and use a 10x. So that's another advantage that we have. So the Sapphire chip is very the basic where the input volume is 25 microliters. So you just prepare your master mix as usual with the primers, probes, nucleotides, the buffers, everything, and prepare the mix and add to the cap, open the cap and add it, and it creates 30,000 droplets. So the maximum the number of droplets are the partitions that we have. So the best would be your efficiency, because you are splitting much higher, so that the idea is to trap a single copy in a single partition, so that if we can count the number of partition which lights up at the end, we know exactly the number of targets. So this is, and also here the droplet volume is validated to be 0.59 nanoliter. So that to the accuracy of the technology itself, I show how it is done. And as I mentioned, 4 samples can be run in the chip, per chip, and you can run 3 chips per run in the machine. So which is like 12 samples in one go. And the limit of detection here stays at a 0.2 copies per microliter of your sample. So that's at the highest sensitivity with the 5 logs. So this is going to be a disposable consumable for you. So it comes in a pack, unpack it, remove the white caps, because this is where your sample goes. And this blue one has actually a filter, because the partition is done through air pressure. So when the chamber is pressurized, so the flow is from the white side to the blue side, which allows the partition to happen. And this is all done in the geode system. The next chip is the opal, which has 16 samples per chip, and here the volume is low. Once you have an optimized system, then you can reduce your volume also. If you screening requirement, then you can do a 7 microliter, and here the number of droplets is 20,000 per sample. So here again, just open, add your sample, ready to go. So there's no user interference, there is no multiple steps involved. So this is the strength of the Stilla technologies. And the Ruby chip that I talked about, so here it has 16 samples per chip and up to 17,000 droplets per well during the sample analysis. So this is how you fix the chips. So it's 3 chips. Here 3 chips of sapphire, 3 chips of ruby or opal, looks same. And the process is very simple. So it takes 15 minutes for the partition. So the pressure pump is also supplied along with the system, and the pressure parameters are maintained during the process. After this, it straight away enters the amplification cycles and completes the amplification process. And immediately after the process, then it just needs to be read under the system. I talked about the 6 color, but we also have a basic 3 color system where you can do a 3 target multiplex basically, but as I mentioned, the compensation matrix will allow you to do more dice in these 3 channels also. Yeah, so these are the common dies that are used, but you can see the emission maximum has about 35 nanometers range, and if you can find multiple dies, you can also use these multiple dies for different targets. So using this particular system, one of the publications that we have is a user has optimized 22 multiplex system. So it's currently in the diagnostic area on oncology markers, cancer biology markers. So the system provides such a flexibility to increase your multiplex, and you can ask me to design a multiplex is complex, but we also support the primer designing and the primer and probe designing for the multiplex. I talked about the fluorescence and the visual quality control. So you can see this. We need to also understand how many partitions did not amplify. So we need to also know visually, like when you have the system scanned or the chip scanned, you will find the non-amplified partitions along with the amplified partitions, and then go for the filters. So these are the white spots are the ones which have amplified, but in this case it doesn't show which dye it has. So the next step would be to apply the different filters to identify and read the different fluorescence. Even before that, there are also physical challenges for the chips, because some can also have air bubbles in the process. Some artifacts, maybe a tissue paper, a thread from a tissue or a thing like that. So those will also be applied to, and the system will provide a signal to show that this can be used for your analysis. So either green or orange. Orange will be okay, you decide whether to use it, but red will be, we don't recommend to use it. So this every time the scan will go through a visual quality control for the user to allow whether I can accept this sample or not. And here you can see two big areas. These are basically the separation pillars. The chip is actually a two-layered system, and it's separated by a pillar which divides these two layers for a very specific thickness. And the partition is a single mono-layer partition, so it doesn't overlap the partitions also. Yeah, so this is about, and you can also see this is the area from where the partition will enter the chip, because originally it will be in the tubes, and when the pressure is applied, it starts filling the partitions from one end to the other, and this is very common. And you can read this, and you can filter. Once you filter, you know which filter is applied, which dye is detected in this case. And the software will provide you a complete analysis of the system. So if you are doing a multiplex, you can also see what are the different dyes. You can have a matrix, and you can show different populations of the partitions. So these are clear partitions that there are only 3 dyes here, and this is a mix of populations. And this mixed population will also give you an indication of which are the 2 mixes that you have, or 3 mixes or 4 mixes that you have. But this will be also this will also provide you a mixed partition that has multiple targets. Yeah, so multiple analytical tools for you to understand things better, so manual, automated, but also it will provide you a graphical representation of each dye that you will be using, and at a publishable quality. And it will also provide you with the information of how reliable your data is. If you are looking at a very low copy number, is this a dependent result or a non-dependable, or requires a repeatable experiment? And the software will also analyze and provide you with the number of droplets. So you can see it would be anywhere between 25 and 30,000 droplets, but you can improve over time. And this also provides how many copies are present in each of these samples. So this comes in blue channel, how many copies, in green channel, how many copies are there, and in the red channel. So it also gives exact number of copies of each target which is available in the template. And also extractable in an Excel format for your further analysis. And as I mentioned, multiplex, the designing of a multiplex assay is quite complex when we think about a typical conventional PCR or a qPCR, but we also provide you with some learning for this. So we can use the Integrated DNA Technologies oligo analyzer tool. You can try it, the version, and further we can also offer the Stilla Technologies platform on how you can design a multiplex assay. So these are all available, and with lot more informations related to your TM, your annealing temperatures, your primer dimers, hairpins, all these are provided there. It just takes less than 3 hours to complete your experiment. So if you already have experience in DNA extractions or RNA extractions, that's ready, you are ready to go with the system. And it doesn't occupy much space. It will be a space, this takes a space of Eppendorf cycler, thermocycler, and the scanner is slightly larger than that, and a PC. That's it. And as I already mentioned, the number of chips depends on how many samples that you are going to process. And this, in case there are also labs where the number of samples are very high. So the scanner they use one scanner and they can run 3 geode systems. So you just need to have a machine that continues to amplify, but the reading happens in 20 minutes. So that is also a possibility. One of the very unique character or very unique feature in this system is the droplet recovery. The Sapphire chips, where you can create the partitions, you can complete your amplification, and then after the amplification, you can retrieve your samples. In case if you find a mutation, for example, if you detect a mutation and if you want to validate the mutation again, so you can recover your amplified products, you can do a regular DNA cleanup and use it for sequencing. This is one of the very unique feature that the Stilla Technologies have worked and patented. So this is something which is very exciting for most of the users, because you can do an amplification, you can do a screening, but if you need to revalidate your sample as a quality control, then this is something which you may have to have. Looking at applications, currently most of the applications are on the human part. I would say more than 75% are on the human side, but of course, there are also several areas in the plant sciences and microbial sciences. Food and GMO testing is something which is very common, because there are already markers which are identified for GMO, and this becomes very simple as a multiplex assay. Quality control, trends, mixed species, and also in gene editing. Gene editing, it's common for both, so plants and animal sciences, and also in the infectious diseases. So infectious diseases in general, pathogen detection, and could also be used in the environmental testing. So if you are interested in environmental testing, so these are the areas which these 5 would be the areas of interest for plant genome, plant scientists. So we have publications, application notes, white papers, which talks about a lot of things, because it took almost a decade for the digital PCR to establish itself, because the technology has to be proven to be superior to the existing technologies, and there are quite a lot of publications. This system can also be used for RNA quantification. Of course, there are other technologies, simple technologies available, but you can also use this for the RNA quantification. And as I mentioned, this is just on the oncology side for detection of papilloma virus, HPV 16 and 18, but doesn't mean it stays for oncology. It could be for any type of virus, any type of bacteria quantification. Yeah, let me move that further. And COVID actually drive the digital PCR accessibility, because with so many variants coming very frequently, digital PCR was the technology to adapt the multiplexing. So you get one sample, run it, and you know which variant of COVID this patient had. So that was so fast in terms of adaptation. And also in the, this is just the EVA green based, not the Tachman probe based, in the sewage, sludge cleaning process. Yeah. So plant studies, this is a slide which I just made because it is also very relevant in plant sciences. The major areas are genetically modified plants at the moment, but it is also used in transgenic lion characterization, in the expression analysis, because these are the two major areas at the moment. But it can also be used in expression analysis, the pathogen diagnostics, the plant species detection, and also the copy number variation in the genome. And we also have few number of publications in plants using the Stilla crystal digital PCR system in barley and in the Prepino mosaic virus that infects tomatoes. Yeah, so these are the publications, and we have a lot of application notes available. So I would like you to visit the Stilla Technologies site to learn more, both from the academic perspective as well as to understand what's going on in the plant science using digital PCR. So with that, I come to the end of the presentation, and I will also introduce our training facility. So we have a training facility in Chennai. So if you happen to be in Chennai, just reach us and we will give you a tour, or we will do a short orientation for the system. And this is the image under the microscope of the chip. So these are the channels, the physically where you can see that there is the liquid layer here. These are actually partitions, and it's basically like a pipetting. So droplets, partitions, and the partitions are arranged in the chip. So this is how the chip is done. So with that, I come to the end of the session and open for any questions.
I think that was a very good update. A follow-up lecture from yesterday. Some of the questions what you had has come out from this presentation, especially the recovery of amplicon for other downstream processes. Now the presentation is open for you. Yes, please.
Sir, my first question is like, at one time, how many samples can we load? Will it depends on the chip we use, or what?
Yeah, it's per chip you can load 4 samples.
Per chip we can load 4 samples.
Yes.
Sir, and you were mentioning about 2 bit chip, per chip, and Sapphire chip.
Yes.
So on what criteria we have to decide which chip to use?
Yeah. Yeah, so it depends on the throughput and your requirement for recovery. So if you want to recover your samples, we recommend the Sapphire chips. But if you have optimized, because generally what happens is it takes some time for you to optimize and validate your experiments. And if it doesn't require 30,000, and if you have sufficient target copy numbers, then you can move to the Ruby or Opal chip. To start with, we would recommend Sapphire because it gives more flexibility, more, as I mentioned, it can also be used to recover your samples for further analysis. And then if the number of samples are more, then you can use the Ruby or Opal chips.
In the part of the machine, you said like 4 samples. For the scanner, is it the same?
Yeah, scanner also holds 3 chips at a time. So if one batch of chips on the geode, after amplification, it can go as such in the scanner prism.
So you were mentioning if we have many samples, we can use the machine with
Yes.
the scanner.
Yeah, so typically what happens is the geode takes about 2.5 hours to do the partition and amplification. And the scanner takes only 20 minutes to scan. So the scanner will provide a lot of time. So if you have 3 geodes running simultaneously, and the scanner can continue to scan the samples at the same time. It is not a bottleneck in the process, but geode would be a bottleneck if you have a very high number of samples. Otherwise, we don't really worry about it.
And you mentioned about the runtime is around 3 hours.
Yeah.
So does it depend on the sample? Like if you have only one chip, the duration is that like
No, it's like your PCR only. So you just, yeah, you have your samples and your partition, denaturation, annealing, extension, and the cycles. So we give about 40 cycles in this case, 45 cycles sometime, because especially when we look into the single copy detections, we go up to 45 cycles. Yeah. So considering 40 or 45 cycles, it will take less than 3 hours.
That's a good question.
Yeah.
A lot of potential application for microbiome research, multiplexing especially. When he was making a presentation, that was running in my mind. You have a marker specific for proteins. You have a marker specific for fungi, specific for toothpaste, and specific for green acids. Maybe we have some microfauna also, front fluorophore. And you can really count the microbial load in an environmental sample using DD. Don't you see a potential here? This
And
is one application.
yeah.
I don't think somebody has made an attempt to
Yeah.
do that.
Yeah.
Because in microbiome research, we always quantify the number of organisms in an environmental sample. Most often, the current technology allows one at a time. Either you go for bacteria count or you go for fungal count, but not all of them together in a single analysis.
Yeah.
I see a potential. The only thing is, carefully how to do that multiplexing using different fluorophore with the different markers and a single analysis, because the machine is going to tell you actual count of organisms in the sample. So this is one of the potential applications.
Yeah.
From that perspective, this is a technology which we can really think of, just to assess the microbial load in the environmental sample.
Yeah, and just to add to that, the biggest challenge Dr. Kumar would accept in an environmental sample is the inhibitor.
Exactly.
Yeah, and typically when we have the qPCR applications, the inhibitors play a very important role, because, and that will be eliminated, that the challenge will be eliminated with a digital PCR.
I fully agree with you. And the current DNA nucleic acid extraction procedure, we were using the same technology 20 years back. We always end up getting a lot of the so-called co-precipitating chemicals from the soil system, especially humic acid, fulvic acid. They are all potential inhibitor of any downstream route, not only PCR, even restriction analysis.
Yeah.
It won't work. So now the current tools and current kits are quite improved, and the quantum of inhibitors are below the threshold of inhibiting a downstream reaction.
Yeah.
I'm sure this technology will have an impact in microbiome research in terms of assessing the total microbial count in the environment itself.
Sure.
Thank you very much, sir. For
Thank you
making
so
this
much.
presentation. A good addition to the lectures, what we had us to do, especially the multiplexing part. Okay. So thank you so much. So,
Thank you. Thank you so much.
token of affection, we would like to present you a name. Please come. You can come. No issues. Right.
that creates a very, very dynamic system in the plant, right? The plant is quite green. One fine morning you start seeing some kind of symptoms. So meaning there is a different kind of a niche that is being created there, and what you can imagine, the microbial composition is changing there. Remember, microbial succession and changes in the microbial content is an ongoing process. Nothing is static here. The changes are taking place in a temporal scale and also a spatial scale. We call it as a spatio-temporal scale. There is always a constant flux of changes in the microbial content. So this lecture is structured under 4 major headings. Of course, the last one is a Q&A session. What is plant microbiome? Just to rewind what we have been listening in the last 9 days or 10 days, and who is there, and what are all the tools that is in place right now from different groups, and how we can explore to know who is there, and what do they do. Can we really exploit them for certain important agronomic traits, either to manage some disease or manage the crop, and can we explore the unexplored? I have a case study here, the unexplored microbial diversity for targeted application, and of course the discussion points we'll have. I think this slide was already shown here by Victor, if I remember correctly. This is one of the very famous paper appeared in PNAS. They have analyzed the microbial diversity of the planet Earth, and what they concluded was that 2/3 of the biological diversity is dominated by one particular group of microbe, that is the prokaryotes. So that means they are the real dominant force on the biosphere. And of course, these are all some of the abstracted information from that particular paper. It's quite easy to understand. We have around 40 million bacterial cells in the gram of a soil, and around 4 million cells in the foliar part. And if there is an estimation people have proposed how many bacteria are living on the planet. We know that there are 7 billion human population. And what is the number of microbial count, especially bacterial count? This is already estimated, around 5 into 10 to the power 30 bacterial cells, which is living on the planet. And they represent 1 trillion bacterial species. That is amazing, 10 to the power 12 bacterial species. What I mean, individual bacterial species. And what is known to us is very, very handful, maybe 15,000, if I remember correctly, somewhere I found a characterized bacterial species with a name. And many of them are candidate category. So well-known species are around 3,000 to 4,000. I think this is always under update. So I would say that what we know is very, very handful, and there is trillion and there is few thousands. So that is a difference. And of course, we have not explored 99.999% of the microbial taxa that remains to be that remains undiscovered so far. And of course, being the primary energy provider, the plants are one of the largest habitat for the microorganisms. And it's really a largest habitat. And we know that plants are sessile. They have to live their life in the same place where it got planted. So certainly, it is colonized by microorganisms, both above ground and below ground. And this colonization, I think the entire first week we were talking about that only. It is not the colonization from the microbial side. It is from the plant side. They recruit microorganisms. They invite microorganisms to colonize them. That is the reality. Why they are inviting the microbes to colonize them? In order to do certain functions which plant cannot do on its own, they need the assistance of microorganisms, right? So there are so many examples are there. Please recall all those examples, right? And I don't have any hesitation in saying that the planet Earth is indeed a microbial universe, right? So I make it a clear point, the planet Earth is a microbial universe, right? This slide also you might have already seen in one of the presentations of yours, Raimakers. This reflects the compartmentalization of the microbiome within the plant, both above ground and also below ground. So I already made a point, every plant is a microbial world. Every plant pot is a microbial territory. So every plant is a microbial world, and every plant pot is a microbial territory. So you can imagine a kind of a world, and the world is composed of different countries or different states, whatever you call. And the analogy here is every plant pot is representing a country there, and there the microbes are living. There is a below ground, there is a stem, there is a leaf, there is a phytoplankton, flower, fruits, and interiors, what is called as the endogenous regions. There are microbes. So what I mean to say is the plant is colonized both on epiphytic as well as entophytic region by one or other microbes. There is no plant pot which is free from microbial cells, right? These habitats, they offer the so-called space and growth factors for the microbes to complete its life cycle. So enter into a developmental stages leading up to the production of multiple crops of the so-called reproductive structures. They need energy, and plants offers that energy very comfortably to the microorganisms, here in the case of fungi. And two terminologies you would encounter in microbiome research. One is a core microbiome and another one is satellite microbiome. Let us clarify that, what is that core microbiome and what is that satellite microbiome. I think it was already spoken here by Osramicus. The core microbiome is the one which is consistently present in the plant species, irrespective of their, their origin, their habitat, their season, or their stage. So they are consistently associated. As we progress, you will come to know how to identify the core microbiome. Remember, sometimes some bacteria will trespass their territory. Okay? This is one of the very interesting points. Trespassing means, for example, the rhizosphere microbial communities, the microbiome. They may come out of that sphere and they may colonize a little on the other side, and vice versa. So those organisms, they are called satellite microbiomes, meaning they are not resident of that, but they are the so-called travelers. Okay, they just trespassed and they spend some time there, and they go back or they get vanished from that site. And this is something now it is well recognized, the core and satellite microbiome, right? So that's what I put. Often they trespass their territory transiently, not permanently. Now the question comes, can we predict their permanent settlement? Yes, you can do that. So that is in the time scale, right? In the time scale of the evolution, they may attain the status of core microbiome, but in the life of the plant, in the short duration, these organisms are very, very transient. Okay, so that's what I put it here. The transient settlement, colonization during the seed to seed timeline, and permanent settlement you can expect when the plants in the evolutionary timeline, right? So of course they are aptly called as core and satellite microbiomes. So this picture is only to show you that different plant associated niche, they offer space and growth factors for different microbial communities, and those microbial communities, they comfortably settle down. I'll take one analogy here. The organisms which is adapted on the foliar, what is the connecting point across all those organisms? Anybody can answer this question? The foliar adapted microbiomes, they share one common trait. Anybody? You would find the same trait even the pathogens which is colonizing the foliar part. That is the pigmentation. The pigmentation is one of the traits. It connects to all foliar adapted microbiomes because it is an adaptive trait. Why it is an adaptive trait? Because without that pigmentation, certainly the vagaries of weather, in particular the radiations, can make this organism extinct from the face of the microbial world. So it is an adaptive trait, right? And there is even the rhizosphere adapted organisms also share certain traits, if not all, but most of them they share certain traits. You know what is the trait? Motility. because they have to move towards the root exudates. And how it is moving, this movement cannot be passive movement, it should be an active movement, right? So to facilitate that, they need to have some organelles that assist these organisms to move towards the so-called root exudation origin. So motility is one of the adaptive trait of most of the soil-borne or soil-adapted or soil-resident microbiomes, right? I'll just move on. Of course, every plant pot is a microbial world, right? And this is a kind of count, how many microbial cells are living in a particular plant-associated niche. This based on the information what I could gather from the literature. Billions of microbial cells are there in the rhizosphere, millions are there in the phyllosphere, of course, thousands are there in the endosphere, and a few elite of them can trespass into the plant system and create plant disease, right? So, and many of them, of course, are non-culturable, right? And of course, we know that plant cannot live its life devoid of any microorganisms. Plant need microorganisms to lead its normal life, right? And of course, this microbiome, I think you have been exposed to this terminology, the so-called microbiome phenotypes or microbiome associated phenotypes. They are typically, the handiwork of those microbial community association with the plants, right? And one question normally we ask, what is a major driver of this microbiome association with the plant? Of course, it is not fully understood, but right now we have evidences from the data. Of course, some circumstantial evidences also that clearly says that the host environment is one of the major driver of the so-called microbiome composition. Plant genotype, plant physiology, the healthy status of the plant, and of course the agronomic environment, the cultural practices, every factor the plant is experiencing that might play a role in the microbiome association. Simple analogy, if you want to get rid of a plant disease, you spray a chemical. What you are doing, you are eliminating the microbiome from that. Very simple, right? It might happen with all other microorganisms also, right? So in other terms, the plant ecological performance and the microbiomes, they contribute towards the plant ecological performance and also the so-called ecosystem services. This is my own our own thought process now. We have been debating a lot in our group. The disease triangle, I think the students who represent plant pathology must be knowing. Disease is an exception and no disease is a rule. And the disease is an exception. Why? Because the right combination of at least the three factors should cooperate, then only the disease will come. If anyone is not there, you're not going to get the disease. That is a susceptible host, right? A virulent pathogen and also a conducive environmental conditions. If all the three are present in that particular time point, then you may get a disease, right? The question what I am posing here, we don't have any direct answer for that question. The plant microbiome, can it influence the disease triangle outcomes? I think we all know that it can do potentially by influencing every factor of the plant disease triangle, right? The microbiome can influence the pathogen load. Microbiome can influence the plant. We know it very well because we have, you did a practical on that, the defense gene activation by microbiomes, right? And of course, microbiomes also can create or undo the favorable environmental conditions just by secreting some kind of metabolites or some factors that may change the physiochemical properties of that particular environment. What I'm trying to tell here is microbiome is having the potential to influence the disease triangle or the so-called the disease outcomes. Let's see in what way the microbiome can influence the disease triangle, okay? For example, let us see the host genome, right? And host genome expression can be modulated or can be influenced by the microbiome, right? We have already witnessed it, right? How it can interact with the so-called pattern recognition receptors, right? And in the process, it can activate the immune pathways in the plant system, and it can create the so-called plant resistant to a pathogen attack. That is one way of looking at things, right? And they can influence the pathogenome capabilities, right? What is called as intermicrobial interactions, right? In the presence of a potential antagonist, a pathogen may not perform its virulence function the way it should have been. So this is another concept, right? And it can, influence the so-called environmental factors. For example, they work in the environment in the cooperative manner. They come out with a kind of factors that might influence the host and pathogen at the same time, right? So what I'm trying to tell is each and every interacting partners here can have a direct effect from the microbiome or microbiome associated factors, right? So this is some of the thought process. The effector mediated immunity or effector mediated susceptibility both would be under the attack of microbiome or microbiome associated factors. That is quite possible. And the same way, the so-called MAM triggered immunity or the MAM triggered immunity pathway can be accelerated with the help of the so-called beneficial microbiomes. And the crosstalk between these two is one of the potential factors to keep the plant resistant. And the microbial cooperation and competition for resources with the pathogen, we know it very well in biological control systems. The so-called niche exclusion is one of the famous theories, or antibiosis and microbial antagonism can reduce the threshold level of population and can create a condition for plant to survive. So these are all some of the thought process. Again, you can just go through it. The contribution or the impact of environmental microbiome in the disease triangle is currently very sketchy. There is no clear, clarity on that. Maybe you can have circumstantial evidences and the data sets from different papers. You can stitch all of them together and come out with a proposal. And there are some presumptions here. For example, the microbial succession and the energy transfer in the microbial network is crucial to understand the pathogen ecology and plant disease outcomes. And of course, any disease mitigation strategy, it must encompass the associated biological factors that includes the microbial domain, right? So what I'm trying to tell here is we need to move on from the reductionist strategy. Of course, the reductionist strategy is something which really contributed in the better understanding of the plant disease outcomes. So for example, the so-called Koch postulates, whatever you are doing, is typically under the title of reductionist strategy. What is reductionist strategy? You are isolating the organisms from you reduce the complexity. The isolation is only to meet that objective, right? Because it is a complex world, right? So reduce the complexity so that you better understand what's really going on. That really helped us in proving the so-called pathogenicity of several pathogens. It is very much needed, right? But what is also needed to understand the role of microbiome here is the so-called system biology approach or integrated environmental and microbial ecological centric concept. These two are very much needed, right? So you optimally blend these two, the reductionist strategy, along with the so-called system biology approach, and you will be able to do much better job as a, to understand the disease outcomes or to mitigate the plant diseases. With that, therefore, in my opinion, these are all the four broad objectives if you really bring the disease triangle into the microbiome story. Point number one is you need to identify the microbiome playing a role in pathogen viability and pathogen stability, right? So remember, this is one of the new concepts. You are not trying to kill the pathogen, rather you are trying to reduce its potency, right? For example, the virulence contributing factors can be reduced, or the expression of those genes can be reduced with the help of the microbiome or microbiome factors. So this is one of the concepts to identify the microbiomes or microbiome associated factors that can play an important role in, of course, in reducing the pathogen viability so that the number can be brought down. And also, if number is not brought down, at least the microbiome factors can destabilize the so-called virulence factors so that the pathogen is no longer a potential pathogen. It is able to survive but not able to cause a disease. This is one of the objectives. And to investigate the host-modifying microbiomes. This is one of the hot topic. We have been talking about it. Host-modifying microbiomes means whether a component of microbiome can influence the plant metabolism, right? I think the famous ISR, that is induced systemic resistance, and innate immunity activation, multiple MAMs, they're all coming under the same category. You are modifying the host in such a way that the defense is at the heightened state with a bit of a compromise on the growth during the vulnerable stages of the crop so that the crop escapes the onslaught of the pathogen, right? And the third could be to harness the potential of microbiome assisted crop management. So why it is important here for the accelerated discovery of beneficial microbiomes with a due emphasis on plant interacting traits. It's not only the disease suppression but also other plant trait contributing factors. Other plant trait contributing factors in the sense may be to support the plant growth by producing some phytohormones or to supply the so-called difficult nutrients which the plants are not able to take it up, maybe contribute certain micronutrients, maybe some growth factors, and overall you can have an expectation of a better quality product from the plants. This is one of the observations what we made in our project. Whenever you go for a microbiome application on the foliar part, we could see a substantial quality enhancements in the rice grains, especially the rice discoloration is a very substantial reduction, right? So that means that there is no disease control there, rather it is trying to improve the quality of the product, right? And the fourth one is to decipher the microbiome disease triangle negotiations in a targeted manner so that you develop something. So for that what you need a system biology approach where you try to take plant genome function as one of the traits, pathogen genome function, and also the so-called microbiome genome function, put all of them together and deploy a variety of omics tools to come out with something which is beneficial, right? So you need intramicrobial interactions and plant microbiota interactions, okay? We'll just move ahead of this presentation. This is one point, the plant modifying microbiomes. Several microbe-induced plant genes have been discovered already, and in our group itself, we are handling at least a dozen genes from rice itself. They can be modulated or can be changed their expression with the help of microbiome, right? So the combination of microbe and host microbe interaction is proposed to be crucial for the establishment of the plant microbiota and the consequent plant agricultural and ecological performance, right? So example, for plant extracellular and intracellular receptors. They can be the target for the microbiomes. In the process, the plant defense can be activated, and so that the disease can be suppressed. This slide is showing you the variety of shrub niches in the plant foliar part. I already talked about it. Phyllosphere is the overall the sphere of the plant. It has got several subspheres starting from phylloplane, colosphere, spermosphere, cornosphere, coprosphere, and also anthosphere. There is a specific names for all these things. And this is a contrasting phenotype. I already told within the crop season itself, plant offers a very, very contrasting niche for the microbe to colonize, right? So in other terms, what I'm trying to tell here is these contrasting niches can offer diverse options for the microbes to colonize through a process what is called as microbial succession, right? What is there on the healthy plant certainly would change when the plant is turning into a disease state, right? Couple of more slides on phyllosphere, just to re-emphasize why this study is really needed and what is the current state of affairs, right? Phyllosphere, although we say it is an epiphytic one, let us not forget it also encompasses endophytic regions, right? The leaf endophytes or stem endophytes is also part of this particular phyllosphere. This is one paper appeared in Nature Review Microbiology in 2012. That paper says the total contribution of the phyllosphere in the planet Earth is around 10 to the power 9 square kilometers, and it offers as much as 10 to the power 26 bacterial cells. Okay? And this particular phylosphere is a playground for variety of organisms, and many of them are quite known to us. For example, the pathogens which is attacking agricultural crops, whether it is a oomycetes pathogen like Phytophthora or fungal pathogens like Bipolaris, Magnaprothi, Philosticta, the list goes. All of them are adapted on the phylosphere part, right? And the question is, can we bring that invisible microbial world to something which is visible on the Petri plate, right? What I call microbiome prints, right? It's quite easy and one of the easiest things to do, right? What you have to do is you just plate it on a Petri plate and leave it for about 6 hours and peel the leaf after 6 hours and continue the incubation for another 24 or 48 hours and you are going to see this, right? This is a single rice leaf cut into 3 pieces, placed on the Petri plate, and 48 hours of incubation, and the leaf leaves the trail of microorganisms what was living on the leaf, right? So this is a kind of image what you are going to see. What it really conveys? It conveys that there is a microbial world which is existing on the leaf. This is one of the students' work, right? And what you are seeing here is a typical colonies of microbes colonizing the leaf surface, forming a leaf shape on the nutrient agar medium, right? If this is possible, and it opened up a lot of new opportunities for us to explore the phylosphere microbiome. And what you are seeing on the top is a dicotyledon plant. You see the microbial layer, and what is the color you are seeing? What is the connecting point between the monocot and dicot here? Most of the bacteria what you encounter on the phylosphere is typically colored organisms, right? The color is typically yellow, right? Most often yellow. Sometimes you do get brown, you do get a golden color, right? So it is always a colored organisms, right? That is something I call it as a pigmented bacteria, dark pigmented bacteria which is living on the foliar part. I think some students may be working on Xanthomonas, they must be knowing Xanthomonas is not alone which is having the color. There are so many other bacteria which is living on the phylosphere. They share the same color like Xanthomonas. For example, Pantoia, Spincomonas, and Chrysiobacterium, Curtobacterium. They are all having a dark color, right? And the phylosphere microbiome prints on the culture media takes the shape of the leaf, indicating abundant microbial communities on the leaf, right? So how to study them? It is already, I put it here, culturable microbiome and a total microbiome. To study that, you can go for imprinting technique, that is a classical microbiological tool. You can go for extraction technique, we have already discussed here. You can go for microscopic technique using scanning microscope. And all those techniques would convey the same thing. There is a big microbial diversity which is living on the leaf, right? I skipped this slide because you know much better than this. These are all some of the bioinformatic pipeline what we use in our lab. And of course, you were exposed to a lot more bioinformatic tools in the first 1 week. And this is a typical pipeline to explore, that is sequenced annotation to identification of microbiome on the foliar part. That is, this slide is all about that only. But of course, it reflects a little more. That is about culturomics, phenotypic fingerprinting, phenotypic microarray, and also DNA fingerprinting by box PCR. Okay, so all these techniques would reveal the actual microbial diversity which is present on the plant. This is a pipeline, the culturomic validation of NGS data. I think in the literature discussion, this question was asked to one of the groups by Osrai makers, how the validation was conducted and what are all the data points to say that the metabarcode data is actually being isolated in the culturomic tools. It's quite simple. You have to make a comparison at the OTU level, especially at the genus level. You would see quite a lot of entries. It's quite simple. You have to go for washing, surface washing. We have already discussed here 5 to 6 rounds of washing, and you can go for plating and incubation. You can add a redox dye just to decorate the colonies, and single colony isolation, DNA, 16S, and also identification, preservation and evaluation. This is a typical culturomic validation of MNGS data. This is what I call morphotyping, just to streak your bacteria on a media of your choice and try to document various characters. and I'm sure you would be able to really relate many the same genus. They take the same shape in the morphotyping, and this is also a very valuable tool. It helps many ways to isolate the typical organisms without isolating any duplicates. Certainly this tool is very important so that you don't repeatedly isolate the same isolate over and again. That would add up to the workload. It's one of the very, very important point. You have to isolate something which is unique and not to isolate the same colony again. For that purpose only we use this redox dye addition in the media. With that background, I'll take you to the tour of two case studies, how this phylosphere microbiome has really helped us to identify certain organisms that can be potentially utilized in agriculture as a disease management options, right? The first case study is the phylospheric endophytic microbiome, exploring the phylospheric endophytic microbiome. I make it point very clear, endophytic bacteria or endophytic microbiome, which is colonizing the foliar part of the plant, particularly the leaf, right? We had several research questions to address. The number one is, what are the major drivers of leaf endophytic microbiome assembly and composition? Is there any rice leaf specific endophytic, core endophytic microbiome? Can we find genetically identical microbial communities in the contrasting climatic zones, the core microbiome? And can we harness the potential of the core endophytic microbiome for plant health management? These are all some of the broad, basic as well as the applied objectives what we had in this project, right? Of course, to address some of the larger questions, what are all the major drivers of the assembly and composition, genotype and environment, or their interactions? Can we harness them for microbiome assisted crop protection? For that, what we did, we ventured into 3 different locations in the country, starting from Palampur, somewhere in this sample is from Palampur, then perhaps this is from Almora, this is from Jharkhand. They were the places, and the 2 types of rice genotypes were planted in that location, and we deployed the tools like culturomic as well as the so-called metabarcoding tools to explore, and the same sample was bifurcated, and one set has gone into metabarcoding and another set has gone into culturomic tools. Remember, the sample is same, it has gone into different analysis, right? And this is a story, and these are all some of the statistics which I really don't want to bother you, but what is the most important point here is the number of species what we observed in those samples. You can see here the number varied from as low as 71 species right up to 173 species. So this is a data set, and what we found was the meta-barcoding assisted profiling of rice endophytic microbiome. It revealed abundance of at least 4, the major classes of bacteria, that is Proteobacteria, Bacteroidetes, Firmicutes, and Actinobacteria. They were very consistently present in the higher taxonomic order across all the places, right? This is a Bray-Curtis analysis, and this graph is very important because this graph is something which reflects the microbiological validation of metabarcode data, right? And just have a look here. This is the metagenomic or metabarcode decoded OTUs, and 2 varieties of rice, PB1 and PPT5204, basmati, non-basmati, and this is a cultured one. And that also reflects our inability to isolate all OTUs what we encountered in metabarcode data. That is quite understandable because you cannot culture all the bacteria, right? So, and what is more striking here is the same color whatever you are encountering in both the sets is the one is validation, right? The one which is there in the metabarcode is also found in our cultured one, right? So this is, of course, this is a genotype-wise comparison. This is what they call location-wise comparisons, right? Both metabarcoding as well as the culturomic tools. So what does it really convey? It conveys that as long as you are addressing the same sample, deploying variety of technique, you are getting better, what do you call, integrated data emerging from the data sets, and you are in a better position to explain what is really going on. So the crucial aspect, some of you may be doing, or all of you may be doing this kind of work in future, ensure that your sample processing is one and analysis are multiple. If you are doing two different sample processing, probably you may not get the kind of correlation between a different type of analysis, right? If you want to have a correlation, certainly the sample should be a homogeneous sample processed by the same person at the same time. So all those things are very, very important here, right? And the principal component analysis indicated the key role of agroclimatic zone in shaping the endophytic microbiome assembly and composition. For example, we found certain orders, for example, Burkholderia, Enterobacteriales, Pseudomonadales, Rhizobiales, Spincomonadales, and Xanthomonadales. They are forming the core microbiome of rice, right? Of course, it is a class level. And we have tools to, identify the core microbiome. So the microbiome analyst program, Victor was talking multiple times on the dais. There is an option for identifying the core microbiome. You run your sample. In that particular option, you start getting a graph like this. What it really conveys to is the so-called core microbiome of your samples, right? So this is at genus level. We found, with respect to that particular sample set we got, but if you put all of them together, this is what you are going to see. The bacterial genus belongs to Acidovorax, Actinobacteria, the Rhizobium complex, Ariomonas, Bradyrhizobium, Burkholderia complex, then Enterobacter, Pantoia, it's very important, and Pseudomonas, Spincomonas, Ctenotrophomonas, they are the core endophytic microbiome of rice leaf in our study, right? So this is a kind of validation part once again. After doing the metabarcoding analysis, we went into the core collection. We got around 283 bacterial colonies. They were characterized, and it has fallen into 163 discrete species, and they 163 discrete isolates based on the box fingerprinting. And this 163 has fallen into 78 bacterial species representing 31 genera. So this is from one particular set of sample, right? And of course, they all already uploaded in the GenBank. It is quite, it's available there. So now this is some of the statistics, Shannon diversity index and other parameters. I'll just skip these slides. And this is what the comparative metabarcoding data, comparative culturable endophytic microbiome data speaks. We could find even a specific bacterial association in different sample sets, and I would call them as which has come from the environment that is having locally adapted organisms. They colonize those plants. This is a location-wise comparison. We could see that there is a lot of common organisms between the two locations, Palampur and Almora, but we could find very little comparative entries when you are going away from the. Let us not forget Palampur and Almora, they share the same ecological characters. They are they are mid-Himalayan range. And if you bring this Hazaribagh there, it is somewhat plain. So we could see very little commonality between these two sample sets. So what does it really reflect? The reflection here is the environmental factor has got a major impact in the microbiome composition and the diversity. This is quite clear, right? And surprisingly, we could not find even a single common entry between Almora and Hazaribagh. Remember, this is from the cultured one. Of course, there might be some common entries when you're talking about the uncultured one, but this is the cultured one. Yeah, this is, I already talked about it. I will skip this slide. This is a culturomic validation of MNGS after OTUs. You just put all of them together in the single slide using the same color code. It will give a better visualization of what is common between these two data sets and what is uncommon between these two data sets. It's quite easy to do that. Simple to the graphs, right? Yeah, this paper we could publish in the microbiological research 2 years back. And this paper is already there. If you want to read, you can just go through this paper, and that will give you a much better idea of this particular work. Now I'll move on to another area. Is there any specific core endophytic microbiome? We did find some core specific rice associated microbiomes, not only from our work. We just tested with the data which is presented by so many other workers who is working on rice. These organisms are usual suspects what you can encounter in rice, right? For example, Acidovorax, Acinetobacter, Rhizobium complex, Oriomonas, particularly the Enterobacter, Pantoia, Pseudomonas, Spincomonas particularly, and Stenotrophomonas. They are all ubiquitous on the rice leaf endosphere. Key finding of our experience on this particular exploration of leaf endophytic microbiome of rice, which is grown in diverse agroclimatic zone in our country. In 4 bullet points, the meta-barcoding profiling of rice endophytic microbiome, it revealed abundance of the 4 major classes, Proteobacteria, Bacteroidetes, Firmicutes, and Actinobacteria. The Breaker-Tis PCOA analysis clearly revealed the agroclimatic zone has got a major impact on the composition of this leaf endophytic microbiome, and we could narrow down at class level, order level, the core microbiota, that is Burkholderia, Enterobacterial, Pseudomonadales, Rhizobials, Stincomonadales, and Xanthomonadales, and of course we could find these 4 orders in the cultivated microbiome also, right? Take it forward. What we did, the question is why we are doing all these things. Can we harness the potential of this endophytic microbiome for some agronomic traits here? That is a primary question. We are answerable for certain applications or applied research. So we have taken this disease, the blast disease, as a target here, and the hypothesis here is if you increase the number of this endophytic microbiome on the rice leaf, that might offer a non-congenial niche for the Magnoporthy to colonize. It's a very simple concept. So what we did, we have taken all of them, we have screened them in planta. Remember, this is very, very important. Of course, we did in vitro as well, that is a secreted metabolome and also volatilome. And in addition to that, every organism was tested in planta, right? How in planta testing? It's quite easy. You just spray the leaf, the differential dose. Don't settle down for one particular dose. You have to go for at least the 3 different concentration of bacterial load. You spray them and challenge it with the pathogen and see what's really going on. This is what is called as the tripartite interaction between the plant, the microbiome components, and also the pathogen, right? And what we observed was most often the prophylactic or the preventive spray is something which really do its job far better than the so-called curative spray. If pathogen is already established, if you are doing this spray, very often you end up in a kind of result which cannot be compared with the prophylactic spray. In prophylactic spray, certainly that offers an excellent control of or rather suppression of the so-called disease. This is what we concluded here. And this is what is called as the growth compromise by activation of defense pathways. And this is one of the very startling observation what we encountered. We use rice seedling as a model system here to make it amenable for large scale screening of bacteria, right? Just juvenile seedlings are exposed to the microbiome components and see how the growth pattern varies. If there is a change in the growth pattern, that can be attributed to the so-called defense activation with the compromise of the plant growth. So this is a trade-off that is happening between the defense pathway in the plant and also the developmental pathway, which is always parallelly operating in the plant system. In the process, you may end up in getting a growth compromise on the plant. Let us not forget, this growth compromise never led to death of the seedlings. So obviously that is expected because these organisms are not pathogens. If they are to be pathogens, certainly they come out with a stronger secretions in the form of virulence factors and effectors, and those factors might kill the plant, right? That never happened because they are handicapped for the effector function or the effector secretion or effector biosynthesis. This is something what we encountered in our experiments, right? You see all crazy type of phenotypes, right? And if in a glance you may say that this microbiome is not really doing a good job on the plant, but it is not so. Actually, what it is doing is it is activating the defense pathway, and for that we have quite a lot of explanation supported by the recent literatures And all of you must be knowing the seedling growth inhibition or the root growth inhibition is one of the recognized phenotype of the so-called activated MAM-triggered immunity, right? So this MAM-triggered immunity is something which is the first line of defense against the pathogen. If you can sustain that forever, perhaps there will be a little growth compromise, but at the same time the disease may not come. But we don't want to sustain it forever because it might put a brake on the performance of the plant in terms of yield and quality. So what you have to do is you do this spray in a transient manner, right? Maybe a spray or a couple of spray in the cropping season during the vulnerable stages of the crop when the pathogen is there right in the field, and you create a barrier. You go back to the disease triangle, right? You introduce a microbiome as a transplantation option, right? What it is going to do, it is going to interfere with the plant by activating the defense. It is going to interfere with the pathogen by compromising the plant viability and, pathogen viability and pathogen stability. So these two actions hand in hand would create a condition for the plant to grow in a healthy state. This is what we are contemplating now as a technology, right? So the bacterial colonization triggered phenotypic changes in rice, especially the root growth, is well known now. And of course, we can see the explanation for that, right? So what we did was we have taken half a dozen genes which is known to contribute towards plant defense. And it's written here, CBIP stands for chitin receptor binding protein, CERK stands for chitin receptor receptor kinase, and of course we have PAD4, NPR 1.3, PDF 2.2, and FMO1. These genes are well-known contributor of one or other defense pathway in the plant. So what we did, we challenged the seedlings with the, we introduced the microbial components in the seedlings. In another set, it's a curiosity, we expose the seedling to the volatiles which is produced by these microorganisms, right? And strikingly, the phenotype is somewhat similar. With the live cells, with the volatile, you encounter the same kind of phenotypes on the plant, right? And this is what the results now. At a lower bacterial titer, the transcription of at least the genes, which is put it there, CBIP, CERK, PAD4, NPR 1.3, and PDF 2.2 were induced in rice, and FMO responded at higher bacterial titer than the lower bacterial titer. These the observations. The rice seedlings responded to bacterial volatile by activating these defense genes as well, right? And of course, a slide I just wanted to show you the microbial volatiles. They have rich in different chemicals, and these are all some of the chemicals what we encountered here in the volatile of these organisms. And this is ethyl hexanol from Chrysobacterium, right? Now, one of the objectives I flagged in the very early on in my slide, can we find the unexplored organisms which can be utilized in agriculture? So if you look at very carefully in the so-called the commercial domain, the usual suspects like Pseudomonas, Bacillus, Trichoderma, they really dominate the so-called commercial domain. And 1 trillion bacterial species are already there in the environment, right? So why only few? Why can't we explore something which nobody has explored? So from that perspective, what we did, we settled down for one organism, that is Chrysobacterium. And the name Chrysio is a Greek word for gold, right? So meaning the colony looks like a gold, right? So we, of course, you can take each one of them and go for a validation. But for want of time, we took only one Chrysobacterium. And when we get into the literature, what we found was it's a very interesting organism. It belongs to the Flavobacterium. It gives a very sweet smell when you open the Petri plate, right? It's a Flavobacterium. It emits a sweet smell, very strong flavor, very, very strong pigmented cells. Just open up the slide. This is how the Chrysobacterium looks like, right? A dark, those who are working with Xanthomonas, they would call it as a Xanthomonas. It is something like that, right? But it is not. It is Chrysobacterium, right? So in our collection, surprisingly, we found 10 different isolates of Chrysobacterium. They belong to 4 different species. For example, Trachylium see, we found a Camellia, we can see Culicis, and also Endophyticum, right? The 4 Chrysobacterium species we encountered. And let us not forget, these organisms have never been explored as an option for disease management in crop systems, right? So that is one of the motivations, right? So the dark colored, dark yellow pigmented colonies, and a series of experiments were laid out. And the surprising finding is, I already told it is a sweet smell emitting bacteria, right? In a Petri plate assay, it never produced any toxic metabolite which is having antifungal activity. But what it did, it produced a volatile that is quite strong in suppressing the organisms, right? A strong producer of what you see here is the volatile, right? See, the volatile emission is quite striking here, and it never allowed the fungus to grow at all. And it was subjected to a series of experiments, and this is how the experiments were carried out. You can see very comfortably here. They are able to suppress the rice blast very significantly, and also we could see that several genes are upregulated when this organism is interacting with the rice leaf, right? For example, two of the genes, well-known markers of innate immunity in rice, one is CBIP and also CERK, and these two genes are upregulated as whenever this Chrysobacterium is interacting with the leaf, right? So again, the key takeaway from this experience is the diverse flavobacterial Chrysobacterium we could see on the leaf, and this organism emitted a chemical what is called 2-ethylhexanol, and this chemical we confirmed that it is giving specific antifungal activity against Meganopothe, and it is able to alter the transcriptional profile of rice by activating the genes like CBIP and CERK. And in planted trials, we could see a substantial reduction of rice blast disease when you spray it, right? Now we are investigating this organism. I think the whole genome has been cracked. I think Shanu is working on this organism. The whole genome sequences have been already generated, curated, and uploaded. The data is already there in public domain. I think she is addressing a full-fledged thesis on that. And this paper appeared in 2021 in Biological Controlled Elsevier under the title Rice Leaf Associated Chrysobacterium, an untapped antagonistic flavobacterium displays volatile mediated suppression of rice blast disease, right? It's a key findings. Once again, I just skip it. We have already explored that. The take-home message I just wanted to convey to you from our experience on the total microbiome exploration on the rice endosphere, and also the specific microbial community like Chrysobacterium. It is listed here. Whenever you are addressing a targeted approach for any kind of agronomic traits, don't ignore the ecological part of the organism or the system in place. For example, here the target is a foliar pathogen, right? And if you are addressing a foliar pathogen, it would be wiser to address foliar ecology as a main target, right? Exactly, we did the same thing here. The microbial and pathogen ecology should be considered while looking for a novel microbial agents for targeted application. If your target is a foliar part and the foliar associated agronomic trait, better you go with those organisms. The reason being, they are already adapted there. They have the ways and means of surviving there. They know the place very well, right? And if you put on that particular niche, certainly they will not get lost somewhere. It will survive somewhere on the foliar part, and it is going to do the job, right? I always call this, I'm not sure how correct I am. If you're taking a soil-borne microbiome on the foliar part, to me it looks like it's a fish out of water, isn't it? So the other way also true. If you're putting something on the foliar adapted, putting in the soil, they would find a completely different place for its survival. So this is something we need to debate, because the ecological component or ecological factor should be considered before we promote certain organisms for some targeted application. What I mean to say is there is no single bullet for all solutions, right? For foliar part, let us address foliar ecology as a main target, right? This is one of the take-home message. And the microbes adapted on the phylosphere or phylospheric endosphere are unique, as reflected in their color and their character. They are very prolific for volatile production. And I see a reason for that, because volatile emission could be one of the adaptive trait of these organisms to prevent any microbial antagonism, right? At a small scale. For example, these bacteria live on the leaf as aggregates, right? They never live as a solitary cells. It cannot. They live as an aggregate, what is called as, in a while, a biofilm kind of thing, right? And remember, in a metabolically active state, the metabolic byproduct is volatile. And this volatile might deter some of the other organisms from nearing to them. So it's a kind of protective environment, right? So not only that, these organisms, they adapted there, they found ways and means to withstand the harsh intra-day vagaries of weather. For example, the early morning, the rice leaf experienced a cold weather. Right after 6 hours, there is a hot sunlight is going to heat up the leaf, right? And in the evening, again, the cycle repeats. And they are vulnerable for a variety of abiotic factors in a single day, right? That means they are exceptionally adapted organisms. There is no insulation whatsoever, whatever insulation they are doing on their own, right? That's what the aggregate is, one of the proposal by the Lindos group. And volatile is another factor that might contribute to their survival, right? And of course, the foliar microbiome members are very unique. Most often they are dark pigmented, prolific for volatile emissions, right? And of course, the foliar microbiome specifically interact with the plant leaf and activate innate immunity by inducing the so-called MAM-triggered immunity genes, right? So these are all some of the take-home message. With that, it's the team who is working day in and day out on these concepts. It's all their contributions, and whatever I present here, thanks to their the hard work, whatever they are doing. They are traveling all over the country for collecting sample, analyzing, right? And with that, I really thank several funding agencies. And I close this lecture by making a statement, one of the famous statement by very famous, geneticist, right? Nothing in biology makes sense except in the light of evolution, right? So that also includes the microbiome as a component of biosphere, okay? With that, I will open this presentation for some kind of discussion. This is a last lecture directly connected to the theme of this training workshop, that is plant microbiome. So from here, we will move to some other topic and we'll have something else to discuss. Yeah, thank you. Put on the lights then. Yeah, any interaction? Yeah, most welcome. Yeah. Yes, please. Yeah, adapted. Yeah, yeah. Yeah, excellent question. See, unlike a soil environment that is quite heterogeneous in terms of soil composition, in terms of the so-called root exudation, soil edaphic factors, it's extremely heterogeneous, right? Even in a single day, you can see the variations. For example, you irrigate, after 3, 4, 6 hours, it dries up. So it's a very dynamic place. But when it comes to phylosphere, whether it's a dicot plant or monocot plant, in a tropical environment I'm talking, right? What we encountered, a strikingly similar set of microorganisms, both in dicot plant and monocot plant. We do have projects on pomegranate. My student Vinod is working on that. And if you see those organisms, you find the same kind of usual suspect which is playing a part on the phylosphere. For example, you will find Pentoya, you will find Spincomonas, you have Methylobacterium, Microbacterium, Bruendemonas, and Burkholderia. So what we are concluding is phylosphere is offering a kind of a niche for a similar kind of microorganisms to survive. They are connected by the same point, the dark pigmentation, right? So I don't have experimental evidence to say it very strongly here, but I may not be surprised the phylosphere adapted organism in rice, it might work in another phylosphere, for example, pomegranate, it might work. But it has to be validated. Yeah. And we have some evidences also. For example, we have a project, another project, we are trying to convert this information into a technology, and where the industry is also involved. The industry is trying to expand the profile of crops away from rice. And they also report to me that it is also working in tomatoes and chili, onions, and wheat. So this is going on. That means we are getting the story connected. So phylosphere as a somewhat a homogeneous atmosphere, except for the volatile profile. You have the same kind of chlorophyll, you kind of same kind of environmental factors, intraday vagaries of weather, it's all same, right? So it might, offer a kind of similar environment for the organisms to survive. Right? That's the reason you have a xanthomonas in pomegranates, xanthomonas in rice also, right? So that is the story, right? Yeah. Any other points? It's a good question, Ashwini. Yeah. Okay, if no more questions are there, we will close this presentation. All right? Tea is already waiting, okay? Okay, thank you all, and let us break for tea now. Then we will come back and we'll have some discussion about a very interesting presentation which US Makers wanted in the course, but he could not do it. But I just brought some information for that. Let us discuss that, okay? Yeah, thank you. That is about publication, yeah. point of time and other agenda, then flight schedule. So you have to cut short that. But nevertheless, I could manage a presentation. This is a little deviant from the core objective of this course, but not too much unrelated. End of the day, you are all working for this only. Agreed or not? Nah? The best satisfying thing for a researcher is to see their paper in a high-end paper. Okay? But to do that, you need to have lot of background work. Okay, we will see. Some of you may be knowing the flow. Some of you may not be knowing. Maybe some of you may be halfway through. But whatever information I could gather and our own experience, I thought I will share it with you. Okay? Title of this presentation is, Art of Publishing Articles in a Reputed Journal. So first day you isolated organisms. Second day you purified it. Third day you evaluated it. Fourth day you sequenced it, sequence analyzed, curated. Yesterday you annotated it. Correct? Now what is left now? Publication. So let us do it now. Okay? All right? Yeah, but it's not that easy. Seriously. Yesterday we received a letter from office. I think Govindasamy also might have received. That letter came from our audit. Very interesting. First time in my career I am receiving such a letter from my audit. All scientists received. Last 5 years, how many papers you published? Year-wise. Okay, number one. Number two question, how many papers got rejected? Okay, third question, what was the main reason for rejection? Right? These three questions, yesterday it came from my office. They gave a deadline of today 3 o'clock to reply. So I will do that after some time. Okay, last 5 years I have to take. Rejection is always a problem and painful, isn't it? The thesis submission date comes next month, 30th. Your paper is still under review. Some anonymous reviewer is reading your paper, making all kind of opinion about what you are, how you did all these things. And end of the day, he types instead of A, C, C, E, P, T, E, D, he says R, E, J, E, C, T, E, D. No? Most often it happens. Yes or no? Huh? Okay, fine. Let's see the some of the small tidbits, rather some fine points, what I could gather from public resources, and some of my colleagues also helped me to make this presentation. Okay, I just titled it something like this: a journey from ideas to experiments to publication. The problem is ideas only, isn't it? It all starts from that only. You have some crazy ideas and try to execute it. You got something and you want to tell the world what you have done. And then you have to endure the process of the so-called peer reviewing. We'll see, we'll run through the slide. Some slides are really interesting and realistic. You will understand, okay? Matching with our own experience. Okay. And on the side, some of the journals we always look into from the microbiological perspective, or microbiome papers. Applied microbiology, microbiological research, scientific reports. These are some of the journals. This is a kind of information what I could share. In the recent times, in the last 5-10 years, there is a spurt of the so-called open peer review journals. What is the meaning of it? Anyone knows it? These journals are publishing your work after the so-called endorsement from the experts in the field, and your science is accepted. It is worth publishing it. Then the journal put it in their URL or their website or whatever you call. So anybody can read that and can refer your paper for their work. This is what is called as, they may cite that. So this open peer review articles, journals, it is only 38 in 2001 to 617 in 2019. Every day it is increasing, okay? And I don't want to read it out, the different publishing houses, there are many. Most popular, I think all of you must be knowing, Frontiers, MDPI, Springer Nature, BMC, and the list goes. These trends are encouraging and signal an increasing interest in the transparency in the peer review process, because in many journals even they display the person who reviewed your work in the first page itself. So that is a very good transparency. You will come to know who has reviewed your paper. If it is accepted, you will, of course, you will praise him and rather appreciate. If it is rejected, you are not going to know who has done that. That's very good actually. So this is some statistics I could bring. 450,000 new articles are published every year. Let us not right now bother who is going to read it. We'll come to that a little later. 500,000 reviewers, 600,000 authors publishing. There is a lot of activities are going on. 600,000 people are writing research papers every year, right? And 19,000 books and 2,000 new books every year. Some statistics I could gather from some of the online sources. Two scenarios are a reality. Let us accept it. What is that? At best, the publication is delayed. Yes or no? At worst, it never published. Okay? We have to accept it. This is a reality. Okay? And of course, the purpose of this lecture is it provides an overview of how to write a well-structured research paper for publication. But of course, some terminologies I brought. Before publishing, you should know what is what are all the steps, what makes a good manuscript, the structure of a good article, and practical tips before you prepare your manuscripts. What is the motivation for publication? This is somebody has done some analysis and they brought out all this 6 point, the first one is the dissemination of knowledge. All of us know that. You are disseminating the data or the interpretation, what you are making, to a larger audience. Okay, so that together we are making the world a better place to live. Isn't it? That's the ultimate goal, isn't it? Why you want to communicate, why you want to publish, right? Career prospects, 20%. All of us do that, no? Isn't it? If you don't have one paper published and one paper submitted, you will not be able to get your degree in IRI. Reality, isn't it? So career prospects. Sometimes we use that publication to get new projects also. Of course, it's a reality. New funding. If you are having a paper in Nature, Science, PNAS, and your proposal, if it is good, then you will get a funding. Because the stamp of your authority on the subject is already there. Somebody has published your work. So career prospects. Fourth one, I'm not sure how far it is true. Maybe there is an element of truth also there to satisfy somebody's ego also. People are publishing. Very interesting. And of course, patent protection and others are different choices. I think thanks to this particular compilation, I could gather all these things. Authors publishing priorities. As an author, certainly the priority for me is, what is a priority for you as an author? What you look forward in the publishing house? Speed. You are submitting the paper today, you want the paper to be appeared tomorrow itself. Nah. That is the worst something, best thing can happen to somebody, no? Isn't it? And the top priorities are speed And of course, standard of the journal also a priority. That's what we always look for, impact factors. And journal reputation also, not necessarily an impact factor. Some journals have a well reputation, very old journal. Impact factor wise maybe less, but the reputation is still there. There are journals, right? So these are all some of the expectations as an author before you put your paper, okay? Speed, standard of the refereeing, and also journal reputation. And of course, editor, board, physical quality, publication service also add up to the process, okay? And journal selection, of course, it is already there. The key factors are impact factor, reputation, access to the target audience, overall editorial standard, publication speed, international coverage, and also open access publishers. These are all the top priorities. You really look forward to all those things. Some of the marginal factors they may contribute is experience as a referee, track record, quality and color illustration and service elements. So these are all some of the points it might add up to the publication, right? This is a very interesting slide. I really liked it. The expectations of the author and the expectations of the reader, quite contrasting. Okay? What the author's behavior, let's see, what he really is. You want to publish more. Yes or no? From one thesis, you want to publish 10 papers. Correct? More paper, you seriously, right? In the number. Peer review essential, other journal function, and wider dissemination. Let's see the reader behavior. Want integrated system. He want to get the information as in a single snapshot, rather than reading too many articles, right? Browsing, quality of information. He wants to read less. I'll just bracket it out. These two are very, very interesting. Author wants to publish more, reader wants to read less. This is a, what should I tell, contradiction. Another thing I'm telling, as an author, you are also a reader. You want to publish more, but you want to read less. Okay, this is a reality. If this is a reader's behavior, as an author, what you should do? You should write less. You should not write more. So, again, what is the point here? Your publication should be as precise as possible so that the reader will not spend too much time in reading and understanding what you are trying to tell. Okay? Remember, in entire publication, these two are the most important trait, author versus reader. Author wants to publish more, reader wants to read less. Okay, that's the reality. Reader's priorities, of course. A paper appeared in Nature, another paper appeared in a journal which you never heard before. Which article you will read? Certainly, isn't it? So, the authoritative quality of quality articles, ease of access, right? The open access journals are living on this particular principle. In a click away, the full article will come. Rapid delivery, convenient format, linking information, low or no cost, and updated information. Somebody should not publish a technology which is outdated already, still publishing a work. Nobody will read that. Yes or no? Right? So, the differences, authors versus reader, once again, authors are journal focused. Readers are article focused. Yes or no? You are journal focused, right? I always tell, sometime our own article we will never read again. The last reading what we are giving is at the time of galley proof stage. Sad reality, isn't it? So, readers are article focused. Right, publish more or read less dichotomy is a reality in the whole game of research paper publication. Okay, that is some of the fact about the so-called behavior of reader and behavior of author. Now we'll move on to actual story. What is a research paper? What are all the structures? What are all the factors you should keep well ahead of your actual submission? Let us see one by one. The structure of a research paper is the classical format. I think you must be knowing IMRDCR. What is that? Introduction, materials and method, results discussion, conclusion and reference. Remember, this is a standard format. Whenever you are writing a research paper, make sure to write these headings, no matter what the journal format demands you after some time. Okay? Remember, whatever format the journals are asking, it will be, the content is same. Okay? It may be jumbled here and there, but otherwise the core content remains same. The core content is IMRDCR. You have to keep it ready. Okay? Scientific writing follows a rigid structure, a format developed over the hundreds of years. You read the paper which is 60, 70 years ago, same structure. You have a title, you have a kind of author affiliation, abstract, keywords, introduction, and materials and method, results discussion, conclusion, right? And some journals, so many small subheadings will come. I'll tell you, funding, acknowledgement, conflict of interest. So all those things will come. We'll see one by one. Of course, many times we also do that. In a paper, unless and until the paper is directly related to your work, you will not read the entire paper. You will read only, many times, the abstract. Correct? And it stops there. Right? And I really appreciate the way the literature moderation was held here. He was Roy Makers, he handled it. And he expected you to read the entire paper, and all of you did that. Correct? So what is experience? How much time you took to read the entire paper? 1 or 2 days? If you sit in a single go, how much time it will take? 2, 3 hours? 2, 3 hours? If you are able to follow what is written there, how much time it will take? 2 hours? Okay, provided you switch off your cell phone, right? That's true. Suddenly cling, when sound comes, then done, gone. Your guide is calling you, then you will vanish from the scene. It happens, no? The people are vanishing from the lecture hall also, the same pattern. The paper can be read at several levels. What is that? Some people just will refer the title itself. Super fast people. And they don't have time. Read the title, they will understand the entire paper. Correct? It is quite possible. Suppose a title on effect of so-and-so chemical on so-and-so trait. Will you read that paper? Suppose the information is known to you already. You will not read. You read the title. Maybe sometime you read the author who has published it. Then maybe you may not tell the opinion, but you will vanish, right? Other may read only the title and abstract, and others will read the paper for deeper understanding. So as a student, in my opinion, you have plenty of time, right? So you have an opportunity to read the entire paper, which is related to your type of work. So take it as a pledge that at least during my thesis project, once in a week, I will read at least one paper, the full paper, right? Why paper reading is important, you know, anybody? I'll make one statement, because the statement is told to me by my mentors. Reading will cut short your workload. Read this, keep in your mind. Reading will cut short your workload. Okay, we'll come back to the same statement later. Now this is a classical structure: introduction, materials, method, result, discussion, conclude, and references. Component of paper in little more detail. Okay. Title. What is the purpose of title? It should describe in 2 lines what is the content of that particular paper. You should attract the readers. No? If you see some papers, recent papers, very attractive titles will be there. You are tempted to read actually. No? Yes or no? Hmm? There are many such papers are there. We'll come to that one. Huh? Authors ensures recognition of the writers. Abstract described what was done. Keywords, that is very important because that might help you to get some citations, because people may search using keywords only. Your paper, if you want to be there in the top 10 list, you should use most appropriate keywords. Okay. Then introduction explains the problem, right? Methods explain how the data was collected. Results describe what was discovered. Discussion discusses the implication of the finding. Because every word is discrete and non-overlapping, and it serves a purpose. It's clear or not? Right. And reference ensures previous work is recognized. Many times I used to wonder, can we write a paper without any reference? Why not? You clearly say that similar kind of work was never carried out in the world. No reference cited. Argue for it. Is it not possible? Yes or no? I'm just asking, yes or no? It is possible, but you need to find where it is possible. Okay? And of course, appendices where you are putting, you might have collected so much of data in your thesis, right? And a paper may not accommodate all of them to showcase. So it goes as a kind of supplementary information, right? So all your papers, what you read, the 10 papers, I think each paper is more than 100 pages. That's what I was told, including the supplementary data, what was uploaded by the author in a single hyperlink, right? So what is showcased as a main article is a core finding which reflect the quality of the work. And you expect that without that, the paper cannot be understood properly. So those things will be a part of the main manuscript. This is a challenge actually. Who should be there as an author? Sometimes misused, isn't it? Yes or no? Yes or no? There are guidelines. There are guidelines in our system also. Mails keep coming. How to qualify a person to be author in your work, right? You include only those who have made an intellectual contribution into the research, right? Most likely your chairperson or your guide, mentor, or the committee who is deeply involved in your program, right? And are those who will publicly defend the data and conclusions, that's a senior author, and who have approved the final version, the corresponding author. Order of the name of the author can vary from discipline to discipline. In some discipline, the corresponding author may appear first also. It all depends, right, who has done the work. Let us get into the title. What is effective title? Describes the paper's content clearly and precisely, including the keywords. Is the advertisement of the article. Let us not forget that. It's something like a movie title, right? Isn't it? Title should be attractive. Do not use abbreviations or jargons in the title. Many times I have seen the isolate name appearing in the title. No need. Isolate name of an organism is typically your personalized thing. It need not be there in the title. You can very well remove that. Okay? And do not use abbreviation and jargons. Search engines, indexing database depends on the accuracy of the title. Remember, the title of the paper is very, very important because they use a keyword to identify the relevant articles. Okay? I brought two titles here, both from our work only. After reading, after many years, I realized the first author title is too lengthy, like a paragraph. It should not have been like this, right? And halfway through, we corrected in the next round of papers, read the title, end of it, bacterial flora in root and stem system. It's reading like a paragraph, maybe half of them abstract. Avoid this kind of titles, okay? I admit it's our own work, right? See, this title is a quite clear, straight. Diversity of Ralstonia solanacearum strains on the Andaman Islands in India. Straight, no confusion, right? So title should be very precise. Abstract, I think you must be knowing. All of you are writing abstract, and many times I really go thesis abstract running into 800 words. No. Since there is no restrictions, you write whatever comes to your mind. Writing abstract is not that easy. You have to really spend time. It should cover the entire work, whatever you have done, entire results, whatever you have accumulated, interpretations, and a decent abstract of your thesis should not exceed 500 words. Keep in your mind. Okay, 250 to 500 words. Okay, and some journals they even further restrict. They will ask you to write in 150 words, 200 words. So that would further complicate. You have to sit and really write, edit, right? The readers can decide whether or not to read the whole article after reading your abstract. Abstract should be as brief as possible. Don't reveal too much. If you reveal too much, they read abstract and stop it. Right? Don't reveal too much. You give only the skeletal information so that you are prompting the reader what? To read more. What is their trait? They want to read less. If you are able to make the reader read more, you are a successful author. Correct? So, success is in your hand only. Abstract should be somewhat, there is a saying, it should be non-obvious. Right? It should not be told everything in the abstract. Many authors write the abstract last. Yeah, we'll come to that later, the sequencing, the sequence at which you are supposed to write the paper. There's a trick. Many times students will come, sir, I have completed the introduction part. What? Introduction is supposed to be written in the last, isn't it? So if you are writing the introduction first, that means you are not following the correct path. We'll come to that later. Many authors write the abstract last so that it accurately reflects the content of the research paper. Okay? Yeah, this is typically is our own work. The abstract, some journals, the abstract is subdivided into different components. For example, aim. Then somewhere you will see methods and results, and the conclusion, and significant and impact of the study. I think this is followed by one of the very nice group of journals from Delhi. Now it is from Oxford University Press, Journal of Applied Microbiology. They follow this rule. Okay. Yeah, sometimes abstract is written something like this in a block, right? Telling the story straight away, right? Without any subdivisions. Introduction. Remember, introduction need not be written on the day one. Introduction can be written just before writing the abstract. Okay, all right. It clearly states the problem being investigated, background that explains the problem, and reasons for conducting the research. Many times the paper gets rejected because of this reason only. They may ask what is the novelty in that work. Right? Within 24 hours the paper come back to your inbox. Right? What? It is not suitable for publication. Right? So introduction is very important. You should really, in a crisp, you have to tell what is the current state of affairs and what prompted you to do this research and what is there in the paper. Don't reveal what is there in the paper there. You only tell in a skeletal manner, this is what you have done. Okay? Then explain, identify the questions, state how your work differ from other work, and identify the questions you are answering, and explain what other findings, if any, you are challenging or extending, and briefly describe the experiment, hypothesis, research questions, general experimental design and methods. I think in the literature discussion very clearly told hypothesis and research questions are not equal. Correct? Keep that point in your mind. Hypothesis, you are making a statement and you are making effort to prove it. If you are not able to prove it, you accept it. Okay? Methods. provide the reader enough details so that they can understand and replicate your experiment. Reproducibility. Your work, somebody should be able to reproduce. If they are not able to reproduce, then it is not a science. Okay? Reproducibility is a hallmark of a clear science. Explain how you studied the problem, identify the procedures you followed, and order these chronologically wherever possible. Explain new methodology in details, otherwise name the method and cite an author there so that the length of the article can be reduced. Include the frequency of observations, what type of data were collected, and be precise in describing the measurements and include error of measurement or research design limits. There are so many elements are there. This is a typical materials and methods. Data collection and sequencing, bioinformatic genome assembly and quality assessment, functional genome annotation of the pathogen. So the materials and method portion is something is very, very important because as a student, most often you will be referring the materials and method only just to know how to perform in an experiment in your thesis project. Okay, it's very, very important. Results, objectively present your findings and explain what was found. Show that your new results are contributing to the body of scientific knowledge. Follow logical sequence based on the tables and figures, presenting the findings to the answer, answer the question or the hypothesis. Figure should have been brief description or legend. I will show you a badly presented figure and very nicely presented figure with our own examples, and sufficient to know how the data were produced. Let's forward. Yeah, this is how the results are presented. You can see here, the results are written somewhere like this. You refer the reference, the particular figure here. And this is another way of putting the table. Yeah, the table. And this is a Venn diagram, typical. Results are presented most often both by tables and figures, right? And the table may be a part of the main text, which you feel, which I already told is an integral part of your main text so that people can understand and follow your paper. If it can be there as a hidden material in the supplementary data, you can still produce there because many times if somebody is reading your entire paper, they might want to read your supplementary data also. So it has to be given. So the so-called non-essential data will go into the supplementary section. The most essential data without which the paper cannot be understood, that would become a part of your main text. Remember, you have around 15 tables. The journal is demanding only or requesting you to shortlist only 4 tables. The remaining 11 tables, what you are going to do? Now you have to select what to be qualified for the main text and what to be qualified for the supplementary section, purely based on the content of your paper and also the importance of the data. If the data is totally novel, nobody else published it, then definitely it qualified to become in the main table, main section. And of course, the figure also follows the same rule, right? So this is a typical way of presenting the supplementary. It is always in the form of a kind of a hyperlink. Discussion and conclusion. You have to be very careful. Interpretations have to be made based on the data what you generated. You should not make a far-reaching, interpretations. I think you must have heard about this story. Somebody was some researcher. He was analyzing a frog, right? He was trying to figure out the behavior of the frog if he do some anatomical changes in the frog. So what he did, he kept the frog and he trained it, come near to me. It came near to him. He recorded how much time it took to come near to him, right? So next experiment, what he did, he removed one leg, kept it there. He called again. The frog came, but it took a little longer time than the first one. Obviously, why? It is walking with only 3 legs, right? So he recorded it took a little longer time. Then third experiment, what he did, he removed another leg also. Now the frog was walking with only 2 legs. Obviously, it took longer time, and he recorded. And third, what he did, he removed the third leg also. Now the frog was walking with only one leg. With great difficulty, it reached him, he wrote. And he removed the final leg also. He was calling it. It never came to him. Then he was so excited. And he made an excellent interpretation. You know what is that interpretation? He wrote, when I removed all the 4 legs, the frog lost its hearing ability. You understood? So, why I narrated this story, your interpretation should not be something like this, right? Your interpretation should be something which is relevant to the topic, okay? So, this is one of the, what do you call, a small tip, how not to make interpretation of a data which is not actual, right? So, come to the main point. You describe what your results mean in the context of what was already known about the subject. You corroborate, okay? If you are finding the same finding of somebody else, you say that, okay, I found something which is also found by somebody else. So I am agreeing with somebody else finding. Indicate how the results relate to the expectations and the literature previously cited. Explain how the research has moved the body of scientific knowledge forward. Do not extend your conclusion beyond what is directly supported by the results. The example just now I told. And avoid undue speculations, right? This is a classical thing. Outline the next step for the further study. Okay, these are all some of the points. This is how the discussion flows. Okay, yeah, this is again from one of our papers only. So the paper is about drought tolerance conferred by endophytic microbiome. So what is the starting point? You just read that. What is the starting word in this discussion? Water. Why water? The paper is all about drought. Okay, then the microbiome linking will come a little later. So this is how we, of course, this paper we wrote 2 to 3 years back, and they appeared in microbiological research. Yeah. Conclusion is, of course, remember, conclusion and abstracts are not equal. Okay, many times we make that mistake, right? So don't do that. Conclusion is the salient finding of the entire work in few sentences, or maybe 1 or 2 sentences maximum. Reference, I avoid this because various journals follow different styles, and you follow what is prescribed by the journal editorial board, you follow that, okay? Yeah? Yeah, but there is one point, avoid references that are difficult to find, and avoid listing related references that were not important to the study. It's very, very important, okay? And reference citation, nothing much, you have to select only those references which is directly relevant to the type of work what you did. And these are all some of the styles: Harvard reference style, Vancouver reference style, and I think there are 5, 6 formats are there. And as a student, I think there are several online supports are there. I think I do not know how many of you are following. EndNote is one. So if you are training yourself on EndNote, in a single click, you can format all your references for the journal, what you are handling. And of course, you have to pay some money to buy that. As a student, I think it's worth investing on those programs that would make your life easy. Otherwise, just imagine you have to correct from one format to another format after rejection. How much of time you are going to spend? Nah. Some journal may format will have no punctuations, no dot, no commas, no colons, no semicolons. It is rejected. Next journal you are aiming, they ask all those things. Then what you do, you start putting commas, colons, all those things. So you can avoid that just by investing your time, your money on kind of online support systems, right? Article submission now. So very, very important. Journal choice. So nowadays, a lot of options are there. Some journal publishing house, they have created a platform, journal finders. They may ask you to give the title of the paper, abstract of the paper, and keywords, and it is automatically running your input, and some kind of artificial intelligence system only it will propose. Okay, matching to your kind of data and the work. These are all the journals that will give you the details of the journal, who is publishing, what is impact factor, how much time they take. So you are in a better platform to decide whether to go or not to go, right? So I don't want to read the different publishing house. I think these 3 are the most popular one. All of us are too familiar. Okay, and certain internal discipline. Please read the aim and scope of that journal first of all. Don't submit just in the face value of the journal. First, read the author's instruction of the journal, because that would cut down the time delay, right? They may ask certain format. If you are not submitting in the same format, within 24 hours it comes back to you, so you would lose a day. Sometimes it may take a week also. So why to waste time? So strictly follow the guidelines what is prescribed by the journal for publication. Read the aim and scope and think about your target audience and the level of your work, and do you have a realistic chance of being accepted. This is something very, very important. Some really a top-end journal, they will not encourage you to submit instantly. You have to take a prior permission from the chief editor. So for that, you have to give an extended version of your paper, right? Extended version of your abstract, sorry. So you submit to the chief editor, he reads it, and he finds there is some substance which is worth publishing, then you will get a mail from him that, okay, it's good, you follow the author's guideline, and they will, it will go for a peer reviewing process. For example, New Phytologist, it's a very good journal for plant sciences, they follow these kind of rules, right? Many journals, such rules are not there, you can straight away publish it after reading the author's instruction. Follow the guidelines, okay? Articles should not be submitted to more than one journal. I think this rule you must be knowing. You cannot submit the paper at the same time to 2 different journals. Why? Copyright violations will come, right? If both are accepting it. Of course, we don't do it, but that is one of the factors. In some journal, they will insist you to give a certificate saying that you have not submitted to some other journal. So these are all some of the small tidbits. Nowadays, this is a serious problem, predatory journals. I think you are also getting some mails, right? So this is one of the recent mails I got in 2022. Every day I get, this is something like this. Very attractive title, you are tempted to even publish also there. Journal of Genetic Engineering and Biotechnology Research, right? It's a typical signature of a predatory journal. What they do, they pick one of your papers, they will put it in the content of your mail. We come across your outstanding work. So why don't you submit a paper to us? We will publish tomorrow itself, right? So don't fall for that. So you just see what they are asking. Very interesting here. There is no peer reviewing at all. So we have been searching for a researcher, scientist, authors who will provide their globally high valuable research work, valuable suggestions, blah, 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 so many things. And we have chosen you to contribute. The manuscript paper can be submitted via email only, right? These are all some of the, signatures of a predatory journal. Okay, don't fall for that. And see the journal, what they are, from where they are publishing. And I'm sure after the paper is communicated to them, they may ask you to pay money also to publish it. That is going to happen, right? Anyway, don't fall for predatory journals. Journal selection for authors from low-income countries. Now, there are some publishing house, they encourage science coming from low-income countries, and some incentives are there, and they may not ask the so-called APC. What is APC? Article Processing Charge. This is an integral part of most of the scientific publication. So, of course, these are all some of the journal selection criteria for low-income countries. Remember, paper is one part of the story, but before submitting a paper, most of the top journals, or most of the journals, I would say, they encourage or they expect a cover letter from you. The cover letter is very, very important. In cover letter, you have to describe what you have done and why the paper should be published in that particular journal. And if you published, what impact it may create for the journal, for the work. So you have to clearly describe, but at the same time, don't make overstatement there. Be gentle and make a statement. This is some of the cover letter. I think I just picked from our own work. It typically contains, the statement of the main point of the work. You describe. Genuinely, you describe what is the current state of affairs and why you did this work and what impact it can create if it is published. Very politely, in a very genuine manner, you just put the points there, okay? Then in the same cover letter, you can even identify a potential reviewer of your work. So you clearly write in the end, with this background, we propose the following reviewers for reading our or making recommendations for our paper, because they have expertise in so and so field. Identify the person, his email, his expertise, right? You have to give at least 4 to 5 a potential reviewer of your work who can comment on your work. And there are some discipline. I think most often we are little tempted to give our own internal circle, so that they may give a positive review of your work. Please don't do it. So because many journals, they know who is your corresponding author in the last 5 years. It may not take much time for them to immediately find, okay, it's better to identify a mix of referees. You can put one also from your circle, nothing wrong in that, but don't put all of them from your circle, right? And put one expert who has really contributed in the same field. So that gives you some kind of, your message, you are giving a message saying that the person who is not at all connected with my work, but he has contributed, he is having the expertise to review my work and make a recommendation, yes or no. So it should be a mix of, your own circle and maybe one or sometimes none, right? Or somebody who has really contributed, and ensure that is a mix of representing different countries also. You need not take a referees from your own country or your own institute. But one rule is there, you cannot take a referee from your own institute currently you are working. That rule you have to follow strictly, right? Yeah, cover letter, highlight the key points, do not bring the abstract or conclusion here. You write in the format of a letter. It's a first person singular you have to use. I hope you understand what is the meaning of first person singular, right? I am submitting or we are submitting. You are talking to the chief editor in that letter, so that he reads it as if you are written to him. The paper is attached here. Online submission is the now rule of the day now. All journals are encouraging online only. The offline stuff is gone now. Online has got different options, very easy. You have to create a username, password. So nowadays, many journals, you end up in getting many passwords and many usernames, and many times you get confused. To avoid all those confusion, a system is getting evolved now, what is called as ORCID. I do not know how many of you know that. ORCID, you create a username and password in ORCID, and that is good enough, because many journals, they integrated the ORCID system in their website. Using that particular username password, you can get into the journal and you can publish it. Even GenBank is also using the same. This is what is called as third-party validation of the credentials. ORCID is a very, very convenient one, right? And articles submitted online, and all the review processes also happened online. Speed up the editorial process is valuable for the authors in low-income countries. You don't have to really make a printout, black and white, color, sending by speed post. Nothing is needed nowadays. It's all through online. Okay. After submission, most of the journal editor will make an initial decision about your paper, just to reject or push into the editorial process. Okay. And most editors appoint 2 referees, sometimes 3, sometimes 4 as well, right? It all depends upon the journal policy. And the refereeing speed varies tremendously between the journals, right? And authors should receive a decision of the accept with the revision, minor or major, or reject. Only these scenarios, right? If a paper is rejected, most editor will write you to explaining their decision. The good journal, they do that. They will clearly write in length, much like the cover letter what you wrote, why that decision was made on the work what you did. Sometimes they may say the kind of data what you produced is not what they call matching with the aspiration of the journal. They will not directly tell that it is a poor quality research. They will really say that research quality is good, but you find another journal where these kind of papers are accepted or published. Okay, they are most often they are very polished and very professional. And after rejection, authors have the option to submitting to another journal, and purely your choice. Okay, yeah. This is a very complicated step. So steps needed for the publication. So I already told it all start from here, idea, then translated into some kind of experimental setup, then you generate data, make interpretations, write a paper. So that is, you just put it here, right? So complete your research first, right? Prepare a good manuscript in the typical format I already told, IMRDCR format, okay? You write that. Now screen a suitable journal using journal finders. Then you submit, wait for the peer reviewing comments. Sometimes the desk rejection. Desk rejection means the editorial team, before sending it for peer reviewing, they just reject it for some reasons, right? So it comes back most often in 2 or 3 days. Then sometimes they ask you to encourage resubmission by making the amendments in the manuscript, and you amend it and resubmit to the same journal. That is quite easy process, right? And if they reject outrightly, you search for another journal, okay? So submit, wait for the peer reviewing comments, and do the revision, and send it to another journal or the same journal who initially asked you to revise it. Then acceptance, proofreading. Sometimes it published online. Sometimes you have to pay some money. That is called as APC plus or APC minus, right? APC means article processing charge. Yeah, this is a peer reviewing overview of peer reviewing process. I think the same thing what I already told in a different format. Paper submitted, confirm of the received, initial decision by the editorial chief editor most often, sometimes rejected, then look forward to another journal. And sometimes they decide to review, assign the reviewer, accept invite, reviewer accept. That's where another very important point. Many reviewers are too busy for reading a paper. They may say, I'm not having time. Then it's the editorial office responsibility to find another reviewer, right? Review completed, revise, accept or reject. If it is rejected, again, the whole process restart once again. These are all some of the publishing tips. Remember, editors and reviewers are looking for original and innovative research that will add to the field of studies. Remember, don't reinvent the wheel and expecting a journal to accept your paper. No, not at all possible, right? So for research-based papers, ensure that you have enough numbers to justify a sound statistical conclusions. For larger studies, it may be better to produce one important research paper rather than number of average incremental papers. Remember, it would be a wise idea to have a good paper from a thesis project like whatever you are doing, or maybe 2 at the maximum, rather than generating too many small papers. Later on, nobody is going to cite all those papers. It matters a lot, right? Many often you may not know how the impact factor is calculated. This is a formula here, and this is very simple. The average annual number of citations per article published, right? And international journal have high impact factor with varying time to publish. For example, in 2021, impact factor of a journal is calculated as follows. A is the number of times an article published in 2019 and 2020 were cited in index journals. B is a number of citable items, right, published in 2019 and 2020. And 2021 impact factor is A divided B. So in numbers, for example, 12,000 citations divided by 150 articles for the 2 representative years, and you calculate that is impact factor. It's quite simple. So total number of citations divided by the number of articles which is published in that particular year. They normally take the preceding 2 years they will take, then they calculate the impact factors, right? Why publishing? I already told you, publishing is one of the necessary steps embedded in the scientific research ever since the science came into being. It is also necessary for post-graduation, doctoral programs, and career prospects. Nowadays, the published papers are also help candidate with admission in PhD programs, right, or even postdoctoral programs. What can be published? It's quite simple. New original results or methods, review or summaries or brief on a particular subject. Many times what I have seen, we don't really write perspectives or letter to the editor. Very few of us really do that. There is another area where you can really publish is you can voice your opinion, right, in a top-end journals, right? So what can be done in that particular field based on your own perception and your own understanding, which you feel that can be the way forward, right? This is one area of, it's a kind of opinion papers, letter to the editors or letter to the journal. That is another option. What cannot be published? Reports of no scientific interest, outdated work, right? And duplications, right? of the previously published work, incorrect or unacceptable conclusions cannot be published. Even in thesis, it cannot be published, right? Yeah. Think about why you want to publish your work. These 4 things, it should satisfy new, hot topic, public interest, and problem solved, right? So just make an assessment of your work, whether it qualifies to get the score in all the 4 points, right? And don't panic, good manuscript, major checkpoints, I already told you, manuscript type, aim, enough data, supporting literature, right? These are all some of the minor checkpoints. Scientific message, smooth flow, formatted replications, treatment, graphs, tables, statistics, flow charts. Graphical abstract is something which has been integrated, just been integrated in the new in the publishing style. You know, you are putting your paper in a pictorial format so that somebody can see it and they will understand the entire work by glancing your image, right? So it can be the place where you collected the sample, methodology, the results, and everything integrated in maybe 5, 6 images with this few dotted lines. You can convey the science what you have done, right? That is called graphical abstracts, right? And of course, ensure that your supervisor is checking the manuscripts, right? Examples, every editor, reviewer, reader wants to receive well-presented manuscript. Remember, some journals, they look into the presentation of your manuscript. Science may be good. If the presentation is not good, sometimes they may reject it, right? Ensure that there are no spelling mistakes, there is no typographical errors, there is no punctuation issues, the images are properly presented with the proper annotation. It gives a proper look. That is very, very important. Presentation matters, right? And these are all some of the, this is a typical graphical abstract of one of the papers. This is again a graphical abstract. This is the presentation, right, of a methodology. So what I put X is it's true, it's not a properly presented one, too many colors. So, and you present it properly so that people understand in a single glance. This is the results presentation. Just put, there are certain type of data, you have to use a line diagram, you cannot go for bar diagram, follow the rule, right? This is how a table should be presented. And present a table with the proper annotation. Never ever present the table without annotation. Why it is needed? Because the table should be self-explanatory. If somebody is reading the paper, they should not get back to the text to see what is what there, right? So they have to see right in the bottom of your table what is described there, especially the abbreviations. Good manuscript, you have to provide all the statistical methods what you used. Is one of the most important criteria in the many journals, right? So these are all some of the flow. Very often in microbiome research itself, you have to use a principal component analysis, and again they will ask you which software, which program, whether it is accepted, correlation coefficient, LSD, that is least significant difference, alpha values, R square, and so on and so forth. These are all some of the minimal statistical parameters a research paper should have. If not all, at least some of them you will be having in your manuscript, right? This is a kind of journal finder, which I already told you. There are many publishing house, they came out with online link where if you click it, that will take you to a kind of window where you put the title, abstract, and keywords and run it for a few seconds, then it will take you to another page where it will suggest these are all the prospective journal for the kind of work what you did, and with the impact factor and the time, the turnover time for publishing, all these things will help you to take a decision, right? What you should check is the manuscript perspective consistently with the journal. It's a very, very important point. Never ever forget that. The formatting what you are doing for the paper is matching with the author's instructions which is prescribed for that particular journal. You please follow that, never ever violate, not even a single point, okay? And all the right conclusions drawn from the results, very important point. You should not make a far-reaching conclusions from the few data sets. Does your work add to the existing body of knowledge? Read the guide of the authors again and again. I'm telling you, many times we fail to do that, right? Many times we fail to do that. For example, short communication versus full-length article. Technically speaking, there is no big difference in terms of value of the paper. End of the day, you want to publish it. Many times, I'm also editor in a couple of journals. So they submit the paper under the title of full-length article. I make an assessment. We have a criteria. If the paper is having only 2 tables and 1 figure, it qualifies to become short communication. Then I give a recommendation, they get annoyed. Another mail comes. No, we have done 5 years work. See, that is not the matter here. Finally, I have to convince them, saying that whether it is a short communication or full-length article, end of the day, how many people are going to read it, that matters. Short communications are even better because it's a short. Readers want to read less, right? Anyway, so read the guide again and again, check your word count, layout of the text and illustration, format of the reference, and in-text citations. Are there too many self-citations? It's another major things are now emerging, right? We cite our own work repeatedly over and again, just to increase our own citation. Please don't do it. If it is genuinely needed, you include. Otherwise, you cite somebody else's work so that, it is, self-citation is something, it is not really recommended, right? Yeah. Then, did the first reader of your manuscript easily grasp the essence? And give it to somebody who is not connected with your work. I always say this. And you give it to an economist. Ask him to read the kind of work what you did and ask his genuine opinion, what you feel about this paper. He will tell much better manner whether this paper is really fit for a common man or not, right? That is the last step you have to do before publication. Give it to somebody who is not connected. The best way is give it to somebody in your own family who is not connected to science. Ask him to read and ask his opinion whether this is something which is useful for the common man. And I am sure he will come out with 101 question which you may not be able to answer. That is going to happen because we use many times jargons which a common man will very, very find it difficult to understand, right? It should be easy to understand language you have to use, right? Yeah, English and write in the following order, figures and tables, materials and method, results, discussion, and skip it. Avoid technical jargons and abbreviation, make concise and catchy title. Yeah, I just skip it, yeah. So, I brought these points mostly from the students' perspective. Many students, they would like to make a presentation in a publication in the high-end journals. So you can do that as long as it satisfies certain criteria. First of all, you have a thorough discussion with your advisory committee, chairman, co-chairman, your own lab mates or advice of the manuscript types. Make sure your supervisor reads it twice and make comments. Identify the journal audience and scope. Find suitable journal online as well as ask your supervisor because he may be having a little more experience than you. And review recently published recent publication in the candidate journal to avoid a desk rejection. If the same kind of paper is already appeared in the last month, there is no point in again approaching the same journal for similar kind of work. It's a very, very important point because eventually they are going to reject it. That decision, you reject it first of all, right? That's the best way forward. Never submit to a multiple journal and follow the publication ethics. It's very important, right? Carefully prepare detailed response, letter of response as well as focus on the language. It's very important. So these are all some of the online things I just thought of telling you. The purpose of this lecture is just to tell you that some of the small details about publishing a work. I'll stop it here. Let us have some discussion. Yeah. Anything I missed out, you can please open up. All of you have to do this as long as you are attached to science and writing thesis. Yes or no? Yes, please. It all depends upon how much of contributions you received from those authors. There is no bar. There is no bar. I think I have seen a paper with 100 authors also. And I have seen a paper with a single author, of course. One author must be there. It varies from journal to journal. And for short communication, I think 2000 words is a top limit. The entire paper should be 2000 words. Okay? And for full-length article, it goes anywhere between 6,000 to 8,000 words. It goes with the number of words, excluding, if I remember correctly, title and abstract. From the introduction to I think some journal they put even reference also, but most of the journal they exclude the reference. Up to conclusion, it should not exceed more than 600 words, 6,000 words, 6,000 to 8,000 words. Yeah. See, who I already put, who should be the first author and who should be the corresponding author and who the sequence of author. If you ask me, you should have a proper communication among all the authors who is part of your work. Okay, you should not surprise them by submitting a paper in the journal and they are, my God, somebody has submitted with my name as author. So that kind of, then what happens, every time he expects from you. That is even more dangerous. Correct, na? So once you given the sweet, the person will long for more and more sweet. So this is a problem. So my suggestion is you be, there are rules. Who qualifies to be an author in that paper? Remember, intellectual contribution and physical contribution should be there. If both are there, definitely they qualify. Either one of there, definitely they qualify. Somebody helped you in your experimentation by giving their bicycle, you don't have to give authorship. You make it very clear, right? So one day you are going on the way, you are got late, then they are offering something, you are reaching fast, then you are giving authorship, that's not correct. Okay, there are guidelines. Please read those guidelines. Ethics of publishing guidelines are very clear, and follow those guidelines. Okay. Yeah, it's very interesting. There are. I am not touching that. The topic itself is a controversial actually. There are ghost authors, you know that. So let us not touch that subject. So authors are the one who intellectually contributed in the work, physically contributed in the work, at least either one of them or both of them. That is for sure. Yeah. It is there who qualifies to be an author. Yeah. As a student, my only submission to all of you, make your supervisor a author, because it's your work. You carried the cross. You did everything. Contribution. So that way, definitely there's a decent way of doing it. If somebody has really contributed in the way of experimentation or intellectual contribution, which you feel that person qualifies to be an author in this paper, you give. It should not be like a give and take. You give authorship to somebody, and the somebody is, you are expecting somebody to give authorship to you in the later date. That is not correct. That is means it is ethically it is not correct. What do you feel? Right? Yeah. Yes. Okay, I didn't touch that area. I'm sorry for that. You are asking that, na? What? Exactly. I got your point. I should have informed. I should have brought it here. I didn't bring it. See, there are two kinds of journals are operating in the world. One is what is called as subscription-based journals. That means that journal is subscribed by some library or some individuals in the world. Subscription means what? You are buying a newspaper in the morning, right? You are paying for that. You are buying a magazine. You are making money, right? So then the journal get a profit from that. Okay, so this is a subscription based where you don't have to pay anything. It is free. Most of the society journals are coming under this category. Yeah, most of them. Not all, I would say. Most of them. But there are some society journals also. They ask you to pay what is called as a page charges. Right. Nowadays, what's happening is most of the journals, especially the online journals, they charge you because nobody is subscribing that. It's not a subscription based. It is a APC based. APC means open access, and the open access you have to pay because your article is in a public domain and people are reading it. So it's a business model which is operating now, and they are asking the author to make a payment for the paper that is getting published online. Right, you may wonder what is the benefit. There is a benefit. The benefit is the paper is in public domain, the full paper. You are promoting science. imagine a subscription based journal, you will not have access to the entire paper. If you visit their to read only the full paper. What is option? You have to pay money. They will ask you pay around $35. Then they will give access, the full paper pops up. Right, that is one way. Another way, your institution library may subscribe it. Subscription means what? They are paying. Somebody has to make a payment. Either you make a payment, if it is a APC journal, or your institution make a payment for a subscription journal, so that the publishing house also should sustain their activity, right? It is a kind of way they should do that. A profit, not, if not profit, at least sustaining their activity, they need some support. So this is a publication model right now. Most of the journals have come out, and some are really doing good. Very thorough, and a poor quality paper, they may not publish it. A paper with poor quality interpretations, they may not publish it. So they are quite successful. And in the process, what happens is your science is getting popularized. End of the day, why you are doing the work, you want to make the entire world to know what you have done. Correct. So if it is only the abstracted format, it is presented in the subscription-based journal, the story is only half told, right? If you want to make the story fully told, somebody has to make a fully announced. You have to make, somebody has to pay for it. So that is the reason why some journals are asking APC. Okay, and it goes very, very high money they ask. Sometimes 2,000 euros, some $2,000. Yeah. Okay, we wanted to have this from the Netherlands team, this particular discussion. Probably that lecture would have been even in a different meaning, different format. But this is what we wanted to communicate to you, the finer details about how research is converted into a publication, right? Yeah, we are not touching another area, that is a patenting. So that is beyond the scope of this training program. Maybe you attend one or other IPR kind of training program, they will tell you why you should not publish and why you should patent. So that is a different discussion altogether. Okay. Thank you very much. We will stop it here. I think we are running out of time now. And we will move on to the next activity scheduled for this morning. Okay. What is the next activity? Huh? What? You got it. Okay. All right. Yeah. So, online viewers, we are formally closing this presentation now, and we will get connected by 3 o'clock for the validation. Yeah. Thank you. Yeah.